All right. Apologies for the drop there. We just need to reset our setup here. For anyone just tuning in, my name's Alex. I'm the multimedia technician on board. In the pilot seat, we currently have Scott and co-pilot's Tyler. Scientists will be jumping back on in just a moment. But we are currently diving on yet another unexplored seamount off of Costa Rica. This is the Tengosed Seamount. And I'll hand it back over to the scientists.
Welcome, bueno, bienvenidos Costa Rica, Latinoamérica y todo el mundo. Bienvenidos al buceo número 533 de SuBastian. Estamos explorando el montículo rocoso Tengosed, mar adentro en Costa Rica. En este momento lo que estamos haciendo es explorando. Estas son las primeras imágenes que tenemos de este montículo rocoso. Tenemos algunos datos obtenidos por un barco hace unos 10 años, con mediciones desde el barco, pero estas son las primeras imágenes y la primera exploración que realizamos. We are exploring a site known as Tengosed here in Costa Rica. We are trying to reach the summit of this mount. So this will be our shallowest dive on the whole expedition. We started at 2,300 meters, and our objective is to get to the summit close to 1,900 meters.
So what are we looking at here?
Estamos explorando el montículo Tengosed. Este va a ser nuestro buceo más somero. Esperamos llegar a la cima al final del día, alrededor de las 2 de la tarde, hora de Costa Rica. Estamos buscando señales de flujo de calor, que es uno de los aspectos más importantes que estamos estudiando en esta expedición.
Can you brief me?
Yeah, we are close to waypoint 3.
Okay.
And we made some collections, all that we want and Tomastos, because the one that we collect
Was lost, but you didn't get one.
Not yet, because this place is really steep.
Yeah.
So we tried that. It's really steep. So when you get to a place,
With like a ledge.
Yeah. You can collect that and maybe the tip of the corals as well.
And what else did you guys collect?
Just a branch of the coral with solantids.
Okay.
And
black coral,
yeah.
So that's it, just
That's
those two
it,
things.
yeah.
Perfect. Take your, I mean, you don't have to take them off.
All right.
Hi, Megan. How did you sleep?
So this camera right here
Yeah.
is obviously going to have everything in front of me, but this camera is getting a lot below. So if there's anything down there that you're like, I want that, just tap me or let me know.
All right.
Okay.
Okay.
And if I see anything interesting, I'll either fly down there or I'll reach out to you.
Yeah. Can you just zoom in this area here?
Let me,
Just zooming, not to collect, just to watch.
closer.
Estamos encontrando bastantes corales muertos, muchas esponjas de vidrio. A partir de hoy vamos a tener narraciones de diferentes científicos y científicas. Vamos a estar en la silla liderando el buceo por periodos de 1 hora. También para mezclar un poco el inglés con el español. Estamos muy felices de tener un equipo muy grande de personas aquí que hablan español y queremos que las personas nos hagan preguntas, que conozcan más sobre nuestro mar profundo. Son pocas las oportunidades como estas, donde tenemos la opción de ver con imágenes tan nítidas y tan increíbles las profundidades de nuestro mar. Ayer fue el Día de los Océanos, solo un recordatorio de que Costa Rica es un país marino, somos 92% mar y sobre todo somos un país de mar profundo. Yeah. Yeah. We are almost in point.
Trying to get as close as I can without that probe.
¿Quiere ese? Ok.
Ese no. Ah, ese es un sitio, esa alimenta. Estos son zoantes, tienen un zoante, ¿no sé?
Sí, eran zoantes. Está muerto también.
Todo tiene zoante.
Hay mucho coral muerto, sí. Debe ser por la zona rocosa, debe ser que se golpean mucho. Tal vez. Estamos explorando una área bastante pequeña de este montículo que es de aproximadamente 20 kilómetros cuadrados. En el buceo hoy vamos a explorar apenas una franja de ese montículo. Empezamos a 2.300 metros, queremos llegar a la cima a unos 1.900 metros. Y vamos a tener un segundo buceo en este sitio para poder alcanzar la base del montículo que se encuentra a unos 3.000 metros.
Bueno, también ahí si se recolecta esta rama de aquí agarraríamos los zoantes además del octo.
¿Lo quiere?
Porque ahí está el octo.
Okay.
Y ahí está zoante, se puede agarrar esa rama.
So if it is possible to collect here.
No problem.
All right.
This is not right now.
All right.
Can you take the other location?
Sorry?
I'm done there.
Ah, okay. Muchas gracias por sintonizarnos. En este momento voy a ceder el espacio a Diva, una persona muy conocedora del mar profundo, y espero vernos mañana. Thank you, everyone. I'm passing the lead to Diva. So have a good day. Thank you.
I won't take it personally that I sit down and do that.
So we are trying to have a piece of this coral.
Do you have
here.
the?
Oh, yeah.
This one. It's okay. This one. Yeah, I said it.
So scientists are just doing handover, kind of talking about what the current plan is and all that. Obviously, when you're not on watch here, at some point you might be sleeping, or you might not be near a screen, and so being able to deliver a nice handover report is really important. Catching people up pretty quickly on what we've done, what we're doing, what we still have yet to do, any problems or changes to the plan. So every time they hand over to another scientist, or you'll hear the pilots do it sometimes, or you'll hear me do it sometimes. That's basically what's going on. It's just a quick little report to the person relieving us, and then we carry on.
Well, good morning, everybody. You've probably heard we have new watch shifts. So now for the next hour, you're going to be joined by myself, Dr. Diva Amen from the University of California, Santa Barbara, and Species, an NGO that I co-founded in Trinidad and Tobago, as well as Dr. Miguel Semedo from the University of Porto in Portugal. So we're just trying to set up for a collection here. Pilots guessing, you've heard we want one of the branches with the yellow zoanthids, please. So on this coral, we can see that there's some yellow sections. Those are actually zoanthids that are covering the coral itself. I was about to say, are you okay there?
I've never had to go that far to see something in before. It was just like that in.
Yeah, I don't know, folks at home, if you could hear Chris was bringing the camera into focus, but it was some furious dial twisting. Yeah.
Yeah.
Oh, so the whole thing is dead. Okay. Okay. So then ideally, I think we want to get this branch because it's got both the white and the yellow zoanthids. Please.
This is the overall picture for hours before we do it.
And if we could, is there any way to get those lasers on? Yeah, just before we cut it. Get a sense of scale. Some amazing terrain so far on this dive. These sheer cliffs dotted with sponges and corals. Some little crustaceans, black corals in between. Really beautiful spot. Not particularly easy for sampling though.
Thank you.
Thank you for the wonderful shot.
That's the best
one
idea.
right there.
Now it's gonna find a laser. Oh, there they are. Okay.
Oh yeah.
Okay, so then
Even if you just like can put it close to the base or something, but
there it is on that little part we're gonna be collecting right there.
okay, that's great. Thank you. Yeah, perfect. I mean, some perspective issues there, but it'll be fine. It'll be fine.
Just a second. All right, so we're gonna get. How much of it do you want?
So I think like basically from here upwards if you can, but basically we're aiming for this branch because it has both the white and the yellow sections. Please. So while we're setting up for this collection, we're at about 2,194 meters depth on Tengosed Seamount. That is a seamount that has never been explored in the vicinity of Dorado Outcrop. It is, however, giant compared to Dorado Outcrop. This is definitely the shallowest that we've dove so far on this expedition. Most of our dives have been at about 3 kilometers or 3,000 meters depth, but today, yeah, we're nearly a whole kilometer shallower on the near summit of this pretty big seamount here.
Did you see that fun shimmer coming out of the coral coral quiver? So when they open it, right, we fill these with water on the way down. So that's the salt water down here replacing the much less salty water inside.
And interestingly, that's the same exact thing that we're going to be looking for coming from the rocks on this dive. So two of the things that we are really interested in seeing are that shimmering water, because it indicates where there is low temperature hydrothermal fluid emanating from the seafloor. And that fluid flow, as we saw at Dorado, is often tied to areas where different species of octopus, or rather the moose octopus species, are found to be breeding. So we're keeping our eyes peeled for that type of shimmering water coming out of the rocks. That looks great. Yeah, thank you. As well as any octopus sitting with their eggs, if we can. Perfect. That was like a pro. So our biology collections on this dive are basically going, it can fill a number of different objectives. So one is that Tengosed Seamount has never been explored before. This area of Costa Rica's deep sea floor has never been explored before. And so many of these animals could be new species or new geographic ranges, given they might be found in Costa Rica for the first time. So we need a sample in hand to be able to confirm those things, and if they are a new species, to be able to describe it. Additionally, on board, we have with us a coral expert, deep sea coral expert, as well as a black coral expert. And there's also some interest in echinoderms. So those may be some of the species or the taxonomic groups that we are targeting in our collections today. However, that doesn't mean that if we see something amazingly wonderful or interesting that we potentially wouldn't collect that also. So with that collection in the bag, we are going to continue on to waypoint 4. We're at waypoint 3 right now, and so that's about a 600-meter transit along this ridge. Basically, it's not really a ridge, closer to a cliff edge. And this terrain is just such a sharp contrast to what we've been seeing on earlier dives. Dorado has had Dorado and Fuente Seamount, and the outcrop southwest of Fuente Seamount, the 3 other sites we've dived at, have been much smaller, have had much more gradual slopes, been much more heavily sedimented, whereas here there is this craggy, exposed lava that we're looking at. And that's giving rise to a very different fauna. So a lot of animals that are suspension feeders rather than deposit feeders. So meaning corals and sponges that can attach themselves to the rocks and push themselves out into the current to catch food. But also there's just what seems to be a bit less life. And I guess that's because this cliff face must be a somewhat hard place to live, right? They may be quite strong currents that rip past here. The terrain is very steep, so getting a foothold here may be quite difficult.
I'm going to toss this map back up, but you just heard it's about 600 meters to the waypoint 4. So that's all the way up in the corner there. Let me just crop this down so we got less on there. That's the interface the pilots have to work with. But could I have you zoom out on that map real quick? Because we just heard this was a large sea feature. So waypoint 4 there, about 600 meters away, and we're just going to show the whole feature if we can.
But yes, I will.
Yeah, so you can see we're headed quite a ways out.
Do you know how big this entire feature is?
The entire feature? I don't know.
How tall from base to summit?
Oh, no, I can go ask the folks that were dealing with the map.
No, don't worry.
Yeah,
no, not that important. was just interested. We were just having a sorry. That sounds great. Thank you. That sounds really great. Thank you.
I would ask about that.
Yeah. Because the reason we're having this conversation is that There's essentially seamounts are to give something the formal name of a seamount. It has to be at least, hold on. Ooh, needs to sneeze. It needs to be at least 1,000 meters tall. So for instance, Outcrop, where we were diving on days 1 and 2, is just 150 to 200 meters tall. So very, very small. And that's why it's called an outcrop rather than seamount. So here we were just clarifying that it is actually over 1 kilometer from base to summit, because otherwise it should be called a mound or knoll, I think is the other word you can use. Anyway, so we will clarify that.
A bump on the seafloor.
Pimple. Feature. Yeah.
We're so rude.
All the acronyms.
This is going to turn back into
not acronyms,
like-
synonyms.
yeah, it's going to turn back into the whole Pluto thing, right? That's where we're gonna
Is
get
it
with
a planet? Is it not a planet?
Yeah, yeah.
Oh, poor Pluto.
Yeah, yeah.
It's like poor Tango said.
If you want to put the sit view in the image, so you can see I'm just not steep.
Oh yeah, I can do that.
Also, like this is pretty steep and pretty tall, and these sea cucumbers are like managing to get up there, right? And just be
Yeah.
clinging on for dear life.
You got that one right there.
Yeah.
Yeah. But why to show the view? This is a downward facing camera, so you can see this cliff just kind of goes.
So this sea cucumber is definitely like come a long way. Probably taken him like months.
You remember that the cliffs on the last dive, the like 30-hour
the
dive?
white, the alternating.
Yeah, but there
white,
was like
black
the
cliffs.
slabs up top.
Yeah.
There was a cucumber like at the
I
corner of the
remember
slab.
that.
I was like, oh man.
He's like on the final bit of his journey. Like
Yeah,
I'm nearly
yeah.
there.
Cliffhanger. Like you were saying, though, definitely a different terrain here
Really
than what we've been
different.
having.
Yeah, it's not even like a slope. It's literally like a cliff, like a sheer edge face. And so I imagine it's like really hard for instance, a coral larvae to settle here and find a good spot where it can really like anchor down and grow. Same for sponges. And I guess that's why we're not seeing too many of these larger stalked animals.
Which point? Which point was the 600 meters? That's not waypoint 4. That's like total land.
We are, sorry, so right now going from waypoint 3 to waypoint 4 is 400 meters, you're correct.
Okay, all right.
And then going from waypoint 4 to waypoint 5 is 600 meters.
I'll toss that back up there. There we go. Okay.
Quick zoom on the sponge, yeah. Beautiful. Doesn't look like anyone's at home in the sponge.
Sponge stuff.
But I just love the diversity of shapes and colors and sizes in sponges. Yeah, it's everything from stalked with balls to stalked with weird shapes like this, weird and beautiful shapes like this one, to the little tiny encrusting sponges you can see dotting the rocks behind. Ooh, can we have a zoom on this, please? Oh, actually, I think it's a crinoid. So we're also on the lookout for some of those Anthomastus, which are the pink, the beautiful sort of pink fuchsia corals that yeah, are quite small and stumpy. We did collect one yesterday, but unfortunately, on ascent of the RV, it did get lost. So we're on the lookout for another one today. That is not an Anthemastys, that's actually a crinoid.
Give this 15 minutes here.
Oop, dead sponge.
Just the pilots calling in another ship move there. So get Falcor 2 to stay close enough to us.
Oh, interesting. Area of higher diversity coming up. More life, abundance, and diversity. Bit more sponges. Some more corals. Can't tell if they're alive though. Can we have a quick zoom on all these little white things?
Interesting.
Huh. I am not sure what those white things are. They almost look like polychaete tubes, but like they're not like any ones we've seen before. Yeah, they're exactly. They looked very two-dimensional rather than three-dimensional. Yeah, so we can see some of the ones that I'm thinking of over here. These little calcareous tube worms.
The little ones.
Here,
yeah.
the tiny ones. But then there's these that don't look calcareous. They look.
They do have an open end though.
They do. Yeah.
Yeah.
Okay, so they're probably polychaetes, but a different kind to what we've been seeing before.
Like
Interesting.
little city of them down here.
Yeah, and why all in this one little spot? Yeah, okay, so definitely a sedimented tube polychaete. Yeah, so some of the species of polychaete than we have seen before. But oh my gosh, look at this little brittle star. Oh my goodness. Its like body is so big compared to its legs.
It was cool to see the texture on the worm tube
Yeah,
there.
you can actually see individual sediment particles,
Yeah,
right?
that
And
is
they're
cool.
another one of these coral skeletons covered by zoanthids. So that's a dead coral covered by zoanthids. So not many live corals around here. And there is a little black coral off to the left of that.
That tube was making me think of finding caddisfly larvae as a kid. Yeah, anyone who's never seen a caddisfly larvae,
Sure.
go ahead and look that up. And they build themselves
Gorgeous.
little shells out of gravel or sticks,
Zoom on these, it's
or
just-
some of them will build it out of like little tiny leftover shells from snails and stuff. Yeah, caddisfly larvae are very cool.
So you can see these are zoanthids because it's basically all connected. They have this tissue that completely covers the stalk and then sends out polyps from it. Gorgeous. Just always in awe at the kind of imagery you guys can get. Can we just flick those lasers on for a second? We might have zoomed too much though. Okay, got him. Well, I got one. Perfect. Thank you. Okay, you can continue zooming on the little black coral. So as we zoom in here, you can see one of the different types of polychaete tube worms, calcareous ones just at the top of the screen. There are some little white sponges, encrusting sponges. And then there's this beautiful black coral that we're now able to zoom in on. Great. Thank you. We can move it along whenever you're ready.
Will we be on the return trip?
I mean, that's ultimately up to the chief scientists, but if they will have me, I would very much love that. But also I realized there's a key. Ooh, look at this anemone. It's definitely a balance between having some more experienced individuals on board, as well as bringing in more early career scientists, or perhaps scientists that are new to the deep sea, so that we can really work together and learn from each other.
Check on this one to see
Sure.
if it is
Yeah,
dead
we're gonna
also.
zoom in on this whole thing. And if it's not dead, Oda, sorry, if we could just zoom on both of these, the coral and the.
Less. Is that right there?
Yes, these two. So just in shot right now, we've got a little anemone, little orange anemone to the bottom right, a crinoid about to go off screen, what looks like perhaps a few live corals, a squat lobster just under the ledge on the right, a sea star just under the ledge on the right, and then in front of us, potentially a live, maybe primnoid, and a beautiful homotheid anemone. I think homotheid, but Venus flytraps is what they're nicknamed for obvious reasons.
Yeah.
Nice.
So this anemone is actually attached to an old sponge stalk. And it uses that position to essentially get as much food drifting by as possible.
So I'm gonna say it. Right.
Say it.
We see them as fly traps. But one time someone pointed out to me that without the tentacles, they're shaped like a Pringle.
I have never heard that before.
And now you all have to live with
Yeah.
that. So
I mean, the textures on this are amazing. This is great. Okay, that's great. When you're done getting some good shots, if we could just zoom on that guy next to it. You think? Okay, we'll get a better look for you, Odo. Hold on. Aren't they alive? Woo! So this is a primnoid, right? The one we're looking at right now. What kind of corals? What family? Oh, I don't know. What were you thinking? You're the expert. I can say definitely not bamboo coral. Not a primnoid. Okay. Well, some kind of octocoral. And if we are able to, we would like a snip. You and the zoom over here.
Yeah. So
Is that possible where we are?
I'm gonna ask if you can reach to a test and see if you can reach that. If not,
Any piece you can get your hands on. Snip.
coming to the left is kind of an issue because there's another big coral right there. See just past the outer. If necessary, I could possibly come down below.
So we have been able to confirm that Tengosed Seamount is over 1,000 meters tall. Ooh!
That looks like a baba.
yeah.
Wow, do you have food on the brain, Chris? What's happening?
That's cool.
We nearly.
Doesn't look like it.
Oh.
Yeah, look, it's touching.
Just
No?
But I don't think you can
Oh,
get
it's
the-
fully extended. Okay.
I can come off and see if I can come down low and then just walk from above or from below it and grab it.
Let's try that, and then if it doesn't work, we can look for another one. I think it looks like a gummy, some kind of gummy.
No, I'm gonna move. Do what to do. I know what I did.
Ooh, and there's an Anthomastus, which we also want to collect if possible.
One of the far left.
The little pink, this one.
Yeah.
You think the Anthem is better?
We can do both.
Sorry.
Yeah.
Okay, well, let's concentrate on the Anthomastus first, because that's higher priority.
And then the other one, okay?
Which one do you want? You got two of them.
This
No.
No, which no, that's the same.
one. Ah, okay.
Whichever is easier for you guys, they're probably the same species.
This one's got a better looking right at it.
The pink?
Next.
Said no one ever. Said no biologist ever.
So they got
Geologist
land
probably
on
said
it.
that, definitely. Oda, you sure you want the coral? Because after that coral, we're gonna stay moving for a while, just to because there's a lot of ground to cover. While we're here, we can get it, but then we probably wouldn't collect anything for a little bit. Okay.
Probably, yeah.
So this is an Anthomastus coral. We did collect one yesterday, but unfortunately the sample was lost on recovery. So we are hoping to be able to collect this one to establish. Yeah.
We can collect this one later, but the other one was only alive.
So we'll do both. Well, we're here situated, so we'll just do both, and then we'll move along.
Are you able to get that in the position? Come down the top of the
Yeah, that's
Okay.
well, yeah.
Okay.
So I think while we're sat in this spot, we're gonna try to collect this Anthomastus. If you see any loose rocks, we'll try to grab one. And then we're also interested in that initial coral we had looked at before. And then after that, we're gonna move along to try to cover some ground. But while we're here, let's just do these few things if we can.
Get some wood for it. The laser.
Oh, great. Perfect. Oh, this is a pretty, wow, this is a pretty big one.
Yes, it's gonna be a file box. Your preference.
No, but they were asking which firebox to put it in. Want B, but one of the A's or B, one of these, because they're smaller. Please. Hmm? Yes.
Some fluidity, maybe we'll work out and get some more space. So, drivers. And fire. There you go, okay. Yeah, so then you gotta.
Wait, hold on. I'm
Alright,
sorry. Sorry. So I've just realized the fireboxes need to be prioritized for octopus and microbiology rocks. So,
Okay.
what? What
I
were we
said
gonna
that we
say?
probably would have been octopus and those, but we were looking at, I would think.
Okay. Well then, okay, let's continue then. Yes, sorry. Apologies for the change.
If we come to the point where no, we need that with someone else, it would be hard to get that.
Move it around. Okay.
We're really good approach right there. Go for it.
Man, this is chunky.
You got it all? Good job.
Well
Really good,
done.
really good. All right.
So we can see as we're getting it to go into the bio box, it's beginning to contract its, well, it has contracted its polyps. I think they are able to contract even further than this, but this one is not at the moment.
Don't think this will get too floaty, so once you get in there, it should be okay.
Either of these or
What's that?
flat plates, ERB or just whatever.
It doesn't really, anywhere you can put it.
A little bit too much.
No, it's better, right? Yeah.
It's soft, just.
If you go shoulder down, the other way, up, and shoulder down, so you come out and do this.
Oh, this was great. Well done. Okay, that was great. So while we're in this area, we want to look for a rock. The geologists and microbiologists are saying one that looks like a pillow rather than talus, which I assume means more round than craggy. And like it didn't roll down the hill. So I don't know if you see anything like that. Yes. What, this one?
Yeah, see if you can make loose rocks, the
Yeah, they all look pretty fixed.
blade is not smooth in reference.
Hi. Not that you prepared. Do you see any loose ones? Okay.
Try like that guy right there.
I mean, not any of these broken up dudes because they look craggy, right? So would that one be better?
It's huge.
Okay.
Yeah, that's why. We have to like a small pillow.
We can keep moving. We can keep moving. Yeah, but definitely we're looking for a rock pillow.
She wants the one that we were just
Yes, and then the other coral, please. Okay, so I put it down. We want a pillow lava.
Yeah.
Small. Thanks for coming in to advise. But in theory, one that is more round, right? Okay. Rather than like, okay, like little drops. Okay, got ya. Okay, we'll keep our eyes peeled. Yeah, exactly. Thanks, Maria. Okay, back to the coral.
so.
Wait, was it? It was this one? I mean, I suspect this is the same. Yeah, I think that's probably the same, but.
Below his neighbor.
Oh, Maria just came in as well.
Okay.
But you can also share your
We
advice.
saw some really good ones earlier. Hopefully they're in Sudan. Yep. What are the preference to see if the flow of the salt is low? There's a very first shift.
Like one of these?
Yes. Subdivision.
Okay, sure.
So I know the college
Sure.
of geology nursery is
Violence, did you hear that? We want a branch that has a division in it, please.
So,
Chris, can I see that stick?
So like one of the, like just here would be good, or here, but basically so that it captures a division.
we'll do a test.
Yeah, like maybe right here would be good.
Before we move.
Oh, wait,
Spot,
yes, we
like-
got it before. Yeah, when we were next to the anemone.
There you got money down to sooner. Yeah, so she's fine. All right. You want a shot? Alex?
I actually want a shot. Where'd it go? You bring it over a little bit. These two right here, please.
We're looking for, she wants us to look for a rock that is more rounded rather than like crashed. So, like
Lasers
not
off,
like
please,
that.
yeah. Cool.
No. We can't see one right now. She's just telling us to look for one. Ooh, gorgeous xenophyophore here.
Oh, up top? Yeah.
Also, two little squat lobsters. Some serpulids. Some weird balls on the seafloor. Might be foraminifera.
All right. Thank you. I just wanted that shot.
That's great. Thank you for asking for that, Alex. That was nice.
I just saw him peeking.
Yeah, for sure. Little crustacean goodness.
Yeah.
Yeah.
So here's the best example I can find.
Oh, this is nice.
This shows you a pillar,
Oh,
like
and you
how
can see
it's
where it's like
broken.
fractured. Yeah.
Right, so it shows you that it erupted and had this cooling rind. So if you see something like that, if you see any rocks that look like they fell off of it, it's
ya.
fine. Or yeah, if you see something that's like clearly
Like
an
Maria drew
unerupted-
the
yeah,
drew
yeah,
these.
yeah.
Yeah.
The problem with that is that sometimes
It came
if
from
you
way
zoom-
up there.
Yeah, it came up from up there and then it's really crusted over.
Right.
So often, like I send Amy samples, like I think I got a good intact pillar. She's like, yeah, that was just crushed.
Okay.
So something like that is perfect. And I'm gonna send this to the group.
Yeah, because I was gonna say I'm handing over in like 14 minutes, so definitely the person after me.
We're here, it's here.
Yeah, send a picture to the group.
Perfect. If we can, snip below that. Boom.
Once you have in mind here, a couple of iron oxide trays sticking out of the.
Nice.
Amazing.
Something that's a pillow.
No, okay.
Because well, there's multiple purposes. But pillows will tell us like the origin of the sea, what it was made of, what it's formed.
Alright, and you use that kind of.
If we could just hold it in front of the camera before we stow it, or just get a quick zoom on it, please. Great, beautiful. Thank you. No, that's perfect. Thank you. We can stow it. Yep.
So after this collection, you just want to make some distance on us?
Yes, please. Yeah, we've done a little bunch of collections. Now we're gonna make tracks to waypoint 4 and beyond, please. Unless we see something like earth shattering. And I know that's a relative judgment call, but
With this many scientists on board?
Yeah,
yeah.
Everyone wants something different.
yeah.
Honestly, the geology and biology teams have stayed pretty friendly towards each other, so it's good.
Friendly. Oh my gosh, what kind of cruises have you been on before, Alex? We must say that's one of the most wonderful things about this cruise is that it's been so collaborative and kind and cooperative, and everyone has just been pitching in, helping everyone else, trying to be mindful of everyone's priorities, because ultimately we're all working together to build a picture of this very unique part of Costa Rica's seafloor. So
Yeah.
we get it. Everyone gets it.
It's kind of cool because on this one, every aspect of it really does involve the other aspects, right? Like if you're
Absolutely.
interested in the geology, well, we've learned that the octopus can indicate things about that geology.
Yeah.
If you're interested in the microbiology, that's going to be impacting the octopus. The octopus can tell something about that, and the geology, the water that's coming up, can tell something about that. So yeah, it's really cool to me.
And I think that's what, again, one of the wonderful things about being on a deep sea cruise is that they usually are so multidisciplinary. Like ultimately we are trying to understand this whole connected system, and we need all the different expertises to do that. Oh,
You did. It's just hooked up underneath the handle. There you are.
yay.
A well-placed knot.
Hey, I placed that.
And of course, while we weren't streaming for most of yesterday, do want to hope that everyone had a wonderful World Ocean Day.
Mhm. Are these just a matter of like shoving them down, like they're suctioning?
They will such
Or
an
the-
but they have the little
There is a bit of a
bunting in
screw.
there
Oh, right.
kind of make it harder.
Yeah. So basically we are about halfway on to waypoint 4. We spent we've spent much of this hour collecting, and so after this we're going to try to move a lot and not stop reflections to the worship. So see, I feel the opposite. I wish we should have switched. So we've collected a sea cucumber, black coral, zoanthids, 2 zoanthids on dead coral, an anthomastus, and this live coral that we're just putting away. I don't know. No, there'd be no. Too beautiful, yeah. So something we are looking for on your ship, but also beyond, is a small Dorado, which Ben just put a picture of the website.
Yeah.
But basically we want to I mean, we started my shift on waypoint 3, and we're like still right next week. So, so we definitely want to try to move, and will not be commenting unless it's like.
So just by keeping going.
Yeah, and getting zooms. Getting zooms.
That will be the priority.
Exactly, but getting a lot of zooms, and like when we fly, we have this camera facing forward and this camera facing down, so you can see. But there's not been like a because of so yeah. So we are just getting ready to hand it over. I'm going to be handing over to Sergio Cambronero and still Dr. Miguel Semero. So I'm going to hand over to them shortly, but the priority for the next watch, hour-long watch, is going to be really trying to make some movement along the edge of the seamount while continuing to get a really good look at the diversity of life and geology down here. So thanks very much, and handing over to Sergio. Thanks, Chris. That was great. Those were some stellar collections, seriously. Ready? Oh, I left you my computer. It's a present.
I know, but like if it's something, it's like.
The disco computer.
Yeah, I mean, small little can be good, but challenge, trying to get a bigger one.
Hello, everyone. My name is Sergio Cambronero. I hope you're doing good and enjoying this live stream down from Tengosed Seamount offshore the Pacific coast of Costa Rica. And I will be joining you for the next hour. So hopefully we will be collecting more observations in particularly. We're looking for some geological features. Yes, advancing our way to the summit of this unexplored seamount called Tengosed, which translates to Spanish to English, I'm sorry, to I am thirsty. Pretty particular name. Muy buenos días a todos y a todas quienes nos sintonizan desde Costa Rica y otras partes de habla hispana. Mi nombre es Sergio Cambronero, soy funcionario académico de la Universidad Nacional y director de desarrollo de una ONG que se llama Pélagos en Costa Rica. Los voy a estar acompañando por la siguiente hora. Hemos tenido un buceo increíble, hemos visto muchísimas cosas interesantes en este monte submarino que se llama Tengosed. Y vamos a seguir explorando y navegando hasta la cima del monte. Así que espero estén disfrutando esta transmisión y que se queden con nosotros. Estamos actualmente a 2.172 metros de profundidad y vamos a seguir subiendo hasta la cima del monte, que está aproximadamente unos 400 metros más hacia arriba. Así que pura vida, si nos escuchan desde Costa Rica, por favor, escríbanos por nuestros canales de YouTube y Facebook desde donde nos sintonizan. Y igualmente, si tienen preguntas, comentarios, opiniones, por acá los vamos a estar leyendo. Pura vida.
How you doing?
How you doing, Chris? Pretty good. How about yourself? Good? Nice, nice.
We really died today. Cruising along.
It's cruising along. So special thanks to also the Schmidt Ocean Institute team that's controlling the ROV here in the control room. Chris, Mike, and Jimbo behind the scenes. The one and only Jimbo. They make everything possible. So special thanks to them and all the Schmidt Ocean Institute team. Please let us know your questions, comments in the Facebook and YouTube channels. We will be more than glad to read them and trying to give our best possible answers. Yeah, so we're trying to feel this. Yeah, okay, we got a lot moving this, right? It's almost 10 a.m. Costa Rica time. We're currently diving at 2,171 meters at Tengosed Seamount, offshore the Pacific coast of Costa Rica. We're currently transiting our way to the summit of this seamount, and particularly looking now for some geological features, aka rock samples. So we're on the lookout for that. So please stay tuned as we also see amazing creatures. We previously saw Venus flytrap anemone, and earlier we saw very famous couple, SpongeBob and Patrick. So if you didn't see that, I invite you to go and replay the Schmidt Ocean Institute live streams that they get also stored in the YouTube channel. Ooh. You have that. The one on the left about the geology of the seamount.
Yeah. Oh, sorry.
Oh, no.
Spam call in the middle of the ocean. Yeah, I can. Do you want me to?
Yeah, please. I don't feel empowered to.
So there's a question in the chat about the geology that we're seeing, all the rocks. So what we're diving on today is Tengosed Seamount. It's an ancient volcano that has a relatively flat top. And we're not sure of the age of when the seamount formed, but a nearby outcrop is aged at 23 million years old. So it might be in that same age. It was formed on the East Pacific Rise many years ago. And on this dive, we're kind of hugging the edge of what looks in the multi-beam to be an area where the seafloor has given way. So parts of the rocks have fallen down below, an indication to us of the rocks here having a lot of cracks and fissures in them that allow them to break and fall. What you're looking at here, though, is probably intact rock, as opposed to when we see these areas where you have a debris field with lots of little broken rocks. So these are different types of basaltic lavas. What you're seeing on the screen here, these are rocks that erupted onto the seafloor as molten lava. Oh, look at all the worms on that one. Weird. A type of eruptive lava flow. And one of the things we're looking for is some nice samples we can pick up that will help us date the age of the seamount, and the lava conditions will help us understand its origin. And then we're also sampling interesting microbiology along the way. This is an area where we think that seawater is recharging into ocean crust. That is an insane number of little things. Can we zoom in on that a little closer?
Wow.
What are they? I can't tell if that's biological or mineral. It's very, oh, that's very crumbly. You look at that rock, that's a lot of manganese oxide there. I have never seen this before.
Lots of them.
Have you ever seen something like this?
We see these like
We're looking-
half an hour ago.
Yeah.
Very
This is really close up. It's like crumbly tube.
It's very crumbly, but I just don't know what the white stuff is.
the waters were not sure about it.
Um, it definitely looks like we've got a manganese oxide crust. That's all the black stuff. And there's some sponges in there. That's the gray stuff. But I don't know what the white stuff is. Yeah, they kind of look like polychaete tubes, but they're so flat. There's one that looks like it's maybe.
It looks like they were there, but they're another one.
Yeah, exactly.
Yeah.
I'll take a look at that. Yeah, yeah, now I see it.
Weird. Yeah, interesting. So I wonder if there's just. But how is there enough sediment for that? Okay, weird.
Yeah. Thank you, Beth.
Yep.
Nos hacían una pregunta por el canal de YouTube y que la doctora Beth Orcutt, la líder científica de esta expedición, en conjunto con el doctor Jorge Cortés, nos estaba respondiendo y entonces voy a comentarlo un poco en español. Nos preguntaban sobre la geología de este lugar donde estamos. Entonces, comentarles. Ese. Sí, no sé. Sí, yo creo que sí. Me dijeron como que no era la teoría.
Collections happen at any time. So I think we've collected a lot though. We're only like 4 hours in. So just be mindful of that. At some point we want to get to the summit, but it's fine to stop and collect. There's something, a target species.
Bueno, mientras vemos este curioso octocoral, le escuchábamos de la doctora Beth Orcutt sobre la geología de este sitio, entonces les voy a comentar un poco. Estamos en un monte submarino llamado Tengosed, y esto es un volcán submarino antiguo que no sabemos bien la edad. Sin embargo, por sitios asociados, particularmente el Outcrop Dorado, que son cortezas basálticas de aproximadamente 23 millones de años. Nosotros tenemos la hipótesis de que probablemente la edad de este monte submarino esté por ese rango. Asimismo, vemos acá cómo estamos viendo parte de este antiguo volcán, y todo esto que ven aquí, de acuerdo a la doctora Orcutt y a nuestros especialistas en geología, es lava que salió de la corteza marina, que básicamente aún se encuentra intacta, a diferencia de otras zonas donde hemos visto más diferentes, a veces colapsos. Entonces aquí podemos apreciar algo bastante intacto, y estos son basaltos, lo que se le llaman basaltos, que eventualmente emergieron del suelo marino. Así que con respecto a la pregunta inicial de cuál es la edad de este monte submarino, de nuevo no tenemos un estimado. Justamente esto estamos haciendo en este buceo, ya que es la primera vez que se explora este monte submarino, pero tenemos una estimación de que puede estar de aproximadamente 23 millones de años, debido también a otros sitios que hemos estado explorando y que ya hemos podido obtener muestras. Thank you for your questions, and keep leaving your comments in our Facebook and YouTube channels. We are currently at 2,163 meters and going up to the summit. Our main focus for the next hour is looking for interesting geological features. As we see the fish there, can I take the camera?
Sure. I can go follow if you want. Igual, sí. Quisiera que ya ves que no. Sí, ok. ¿Qué otro? Creo que sí me dice, ya contesté.
Así como vemos muchísima vida que vive asociada al sedimento y al sustrato del fondo marino, también vemos animales que están asociados a estos, pero que también tienen la capacidad de nadar y pasar tiempo en la columna de agua, como este pez que vemos acá, que probablemente sea, vamos a ver, I think we can go.
What's that?
Yeah, I think we can keep it going.
Okay.
Yeah.
Not sure where I'm looking.
Yeah, take it over. I'm sorry.
No worries.
Un saludo a todos los que se conectan desde Costa Rica y otras partes del mundo. Para mí es un honor y un placer estarlos acompañando el día de hoy desde el barco Falcor 2 del Schmidt Ocean Institute. Estamos en vivo y en directo desde el monte submarino Tengosed. Es un monte submarino que es la primera vez que estamos explorándolo. Se explora básicamente en la historia de la humanidad, así que así como yo, espero que se sientan afortunados y afortunadas de estar viendo esto, ya que somos los primeros humanos en ver esto en vivo y en directo. Si se conectan desde Costa Rica y de otras partes del mundo de habla hispana, por favor déjenos saber en los comentarios y por acá los estaremos leyendo.
Looks like we got a pretty dead patch here, man. More relative, there's still stuff there.
Este es el quinto buceo de esta expedición que se llama Octopus Odyssey o Odisea de Pulpos. El objetivo principal de esta expedición es explorar diferentes características geológicas, montes submarinos, afloramientos basálticos que se encuentran en esta área, principalmente con el interés de entender mejor los mecanismos geofísicos asociados a fluido hidrotermal que se han registrado previamente. En nuestro primer buceo estuvimos en el afloramiento rocoso del Dorado, wow, mientras vimos esa neblina cómo se esconde. Y si no vieron ese buceo, el primer buceo, los invito a que lo revisen en el canal de YouTube del Schmidt Ocean Institute, porque vimos muchísimos pulpos. Me atrevería a decir que entre 2.000 a 3.000 pulpos incubando sus huevos. Que también pues estos pulpos están asociados al fluido hidrotermal y por eso es que son nuestros dos objetivos principales de esta expedición, pero también tenemos otros objetivos secundarios como pues, colectar otras muestras de organismos como corales, equinodermos como estos que vemos acá, esta estrella de mar, que son bastante comunes a estas profundidades, y así también importante resaltar que mientras estamos buceando, pues también estamos tomando mediciones de la columna de agua para entender en detalle cuál es la interacción entre la columna de agua y el fondo marino. This is our fifth dive of the Octopus Odyssey Expedition. We're currently at Tengosed Seamount. We're on our way to the summit of this unexplored seamount that we are currently gathering the first observations. And as I was telling in Spanish, I hope that you feel privileged and happy as I am being able to see this live. Probably we are the first humans to ever see this footage and to see these features live from the seafloor. So it is a great moment for the ocean exploration, thanks to the technology and institutions like the Schmidt Ocean Institute, that heavily invest in exploring our oceans. Un saludo a todos los que se conectan. Por ahí veo a algunos colegas. Cristian Pablo desde Costa Rica, pura vida, un saludo. Espero que estén disfrutando el buceo y de nuevo, si se conectan desde Costa Rica a otras partes del mundo de habla hispana, por favor déjenos saber en los comentarios desde dónde se conectan. Con muchísimo gusto por acá los estaremos leyendo. Oh, wow. What's that? Oh. Oh, okay. My man. We are currently seeing, I think this is skate. This is beautiful footage that we're seeing right now. We are double checking what species are we seeing. Just in the meantime, take a look and contemplate this beauty. This is a big one. Can we turn on the lasers, please? So on board of ROV SuBastian, we have lasers that are the green dots that you see on screen that are separated from each other 10 centimeters. So this helps us to assess the size of things. Wow. So this is a pretty big guy. We see some marks and scars, so it might tell us that this is an old. Looks like a kind of torpedo. Yeah. It is massive indeed, probably according to our estimates from the lasers that we have here, more over than a meter. And according to our expert here on board, Beatriz Naranjo, this is a Pacific white skate. We've seen a couple of these type of animals before in our dives, but I will dare to say that this is the largest one that we've seen in this expedition. And actually, that looks unbothered by us. So it is amazing to see this majestic creature as it dwells in the seafloor.
Oh, sorry, bud.
So you can see all the marks and scars on this ray. So this is something that usually we use as an evidence. Yeah, let's leave him alone. I think she gave us an amazing sight. So we're gonna keep going on our way to the summit. Did you note that down? Yes. Yeah. Awesome. Also, we see some comments in our chat that the temperature here is kind of higher than the previous places that we've been visiting. So something that might explain this is that we're almost 1,000 meters up in comparison to our previous dives. We have been diving at around 3,000, 3,100 meters. So usually, as you go deeper, you have slightly more colder water. So in here, it is, being 1,000 meters up from that place, it is expected to have higher temperatures, although this might be also related to other mechanisms that we will then, with the sensors that we have on board our ROV SuBastian, we will take a closer look at that data and see if it actually might be attributed to this, what I'm saying, or other mechanisms involved. We think that this Tengosed Seamount might be a recharge site. And this is part of what we're looking for in this expedition. Not only finding evidence for venting of warmer fluids coming out of the crust, but also if we can find evidence and samples to associate this seamount with as a potential recharge site. So based on previous knowledge and publications from our colleagues on board, the mechanism that we are, oh, that's beautiful. Okay.
You want it?
Yep, we want it. So So while we are on our way to the summit of this seamount, we just stumbled upon this beautiful coral. So we're going to take a closer look, and we might take in a little bit, small sample of it to assess its identity. Many of the things we see right now could be new things for science, new species. So it is important for us, and mainly for our colleagues on board, to get not only this amazing footage, but also to get samples that we can get back on board and take a closer look at it. Sorry.
I just want to make sure that I'm steady.
Yeah, got it.
Keep an eye on that corner right there.
Yeah. Okay.
Okay, we're good.
Can we turn on the lasers, please?
Yep.
So welcome for everyone who's just tuning in. We're live from the Pacific, offshore the Pacific coast of Costa Rica, currently at 2,150 meters at Tengosed Seamount. We just came across this beautiful coral. We are getting enough footage. The green dots you see on screen are separated from each other by 10 centimeters. So you can see the size of this is roughly between 20 to 30 centimeters long. This is a beautiful specimen. I think this is the first time we see this type of coral. So we might give it a go and take a sample. Okay, pero y esos son otros. Okay, listo. So double checking with our colleagues on board, we just confirmed that what we see here on screen are actually two organisms. So we have zoanthids, that are the coral-like critters which have tentacles, and they're host of the bigger animal organism that is actually a sponge.
Yeah.
So
Got a teeny tiny little chitin.
little chitin, where?
Yeah. In the lower right there, there's this tiny, tiny little chitin.
Ah, yeah, take a look at that. Yeah, and there's also beautiful crustacean there.
Yeah, yeah.
Yeah, and beautiful imagery we're getting from our ROV SuBastian. So I think we're good. Give it a go and keep going. Yeah.
It is pretty cool.
Yeah, totally. Can we, like, is it possible to darken the background and just like focus on this? Yeah. So we have a great question from a very, very good colleague in Costa Rica. If that, what are the next steps of all the samples that we take in this expedition? So indeed,
Jorge wants a piece of this one. Jorge quiere un pedazo de la
Okay.
escoba como una suerte.
sí, okay, okay, bueno,
Ojalá la ayude.
el jefe manda, sí, okay. So yes. So please stand by. We were just double checking that we might actually be taking a small sample of this organism.
Yeah, we got
The ray, the skate, yeah, it was incredible, man. Like definitely a highlight. That huge ray, like a meter and a half. So we're gonna. So let's take this little piece here. With this one that's
station and all that stuff as well.
yeah. Let's go ahead. I mean, probably they will fall down, but yeah, let's try and give it a head to
Right?
one of these two branches.
So you can get that into a.
So while our ROV team prepares to collect a tiny bit of a sample of this beautiful sponge that you see on screen, I'm gonna keep following up on the question that we had on our YouTube channel regarding to the process that we do with these samples once we get them on board. So depending on the specimen and the taxonomic group, we take always photographs. So that's the first thing. And that's actually one of my roles here in this cruise. So we have what we call a side photo station, in which we have rulers, we have labels, and a setup of cameras and lights to make sure that we get good photos of the specimen as fresh as we can. So after that, and again, depending on the animal and the taxonomic group, we have colleagues on board that take microbiome samples. So this is for people who are wondering what microbiome means. It's actually the microbes that live associated with these animals, sometimes on the surface or associated, for example, to the outer part of the creature, but also sometimes associated with the gut. So for example, for sea cucumbers, we try to get a little bit of sample from the feces. And this is in order to assess, again, what's the microbial composition associated with these animals. So after that, we usually take tissue samples for genetic analysis. And this is part of what we use to assess and identify the species that usually we do. Yeah, that's good. We usually do metagenomics, and again, depending on that's for the microbiome, but for the genetic identification of the animals, we usually focus on target genes, such as barcoding, and based on that, and if we, okay, can we try like having a
George Matsumoto from Mbari in the chat, thinking that was perhaps a little amphipod, not a tiny little chitin. So potential correction there. I do love zooming in super close and just seeing all the tiny aspects on these things.
But yeah, it can squeeze. Yeah, but I think maybe try to get it from here. And as I was telling, depending on the outcomes of that initial analysis, if we find a new species or something that might give us a hint of a new species or something interesting, we might end up actually sequencing the whole genome, but this is not usual for every sample. So thanks again for our colleagues in Costa Rica, my very good friend Christian, who made that question. Thanks, Christian. I know you're interested into molecular analysis, so thanks again for that question. And welcome everyone who's just tuning in. We're currently sampling this beautiful white sponge that has different hosts, some of them zoanthids. We saw couple of tiny crustaceans, one of them probably an amphipod. Oh, that was perfect. Lovely. Again, and I will take advantage of this moment to let you know that all the sampling involved in this expedition. Oh. Oh, no. It was great. Yeah. Okay. Oh. Yeah, I guess. I mean, if you see if it's possible to fit in there, yeah. So again, as we sample this sponge, all the sampling involved in this expedition has gone through strict processes of permitting with Costa Rican authorities. And even though we take huge advantage of the samples as biologists, and believe me, I take this very personal as well, we try to have a minimum impact on the biological populations at these steps. That's really big in the quiver. Yeah. Okay, so no, it says it's really big. So you don't think it might squeeze it in? I mean, I think it's. You try. And just if you look right there. Just kind of lay that. You think that it will damage it? Well, I don't know if it will damage it. It's just this wide. Yeah. Okay. We can try it though. Want to give it a shot? Yeah. All right. Yeah, I think we want to double check on that. Let me see, because now it's everything else. beautiful specimen, which might be in a sponge with some host, some zoanthids, and some crustaceans. We'll also like to say that these areas that we're seeing right now, that you're seeing on screen, are pretty remote areas. Particularly, this is the first time we visit this place, this particular seamount that is called Tengosed. So the samples that we collect are very valuable to us because otherwise we won't be able to sample. So all of these specimens are going to a strict process of sampling and traceability, and all of them will be deposited into museums. So not only for us to be able to use these samples and advance science, but also for other colleagues from all around the world that might be interested in these samples to have the opportunity to work with them. So most of these samples, I would say the majority are going to be deposited in the Zoology Museum of Universidad de Costa Rica. And also special thanks to all the team at the Zoology Museum that we know that are very, very interested and keen, very supportive for this expedition. So here on board, we're really happy because yesterday was Oceans Day, World Oceans Day, and there was a variety of activities featuring and highlighting the work we're doing here. There were a lot of activities at Costa Rica, also featuring the importance of this expedition to advance our knowledge and empowerment of our deep sea heritage. So for me, also as a Costa Rican biologist, this is a great opportunity not only to advance the science, but also to show the rest of the world and the Costa Rican society that this is our biggest treasure, our deep sea heritage. So this is important because Costa Rica, for those of you who don't know, Costa Rica's territory is 92% ocean, and out of all of that percentage, 60% is located in deep sea waters. So these are areas below 200 meters depth. So we're barely exploring all the extent of territory that we have associated with ocean. Mientras terminamos de colectar este espécimen bastante interesante, una esponja asociada con diferentes zoántidos y crustáceos, veo que tenemos reportes desde Costa Rica, particularmente el equipo del Museo de Zoología de la UCR, así que un gran saludo para ellos y para todos los que nos escuchan y nos sintonizan en los canales de YouTube y Facebook. Si se conectan de Costa Rica y otras partes del mundo de habla hispana, por favor déjenos saber en los comentarios y por aquí los vamos a estar leyendo. ya que para nosotros es importante no solamente avanzar en la ciencia, sino también tener la capacidad de tener un impacto mayor eh, gracias a estas transmisiones en vivo que nos permite tener el Schmidt Ocean Institute. So once we finish this sampling, we will keep going on our way to the summit of this seamount called Tengosed Seamount, which translates to English to "I'm thirsty." Yeah.
Why is that so?
Yeah.
Why is that?
I don't know.
It's a recharging of the water. That's why I want
that might be, yeah. Who knows? Yeah, yeah. So yeah, we have pretty particular names, and that's one of the things that the science party was that we've been talking during this cruise, because we've seen a lot of unnamed features, and there it used to be that anyone who would map these places and identify them, they will be able just to put whatever name they would come up. But nowadays there is an authority that revises this and handles a strict process in order to name geological or submarine feature. Woohoo! Yeah.
Thank you. This was hard.
Yeah, totally. So the robotic arm of the ROV SuBastian is actually being controlled by our experts here in the control room of the ROV, and I gotta say that is pretty impressive to see the skills that they have in order to minimize the impact on the sampling. So special thanks to them for making this sampling process as good as it is possible. So these containers that you see here, in which we place our samples, are also known as quivers, and we use them to place our samples and to keep them protected, to get them safe back on board. So welcome everyone who's just tuning in. We're 2,150 meters. We're on our way to the summit of the seamount, and we see some comments in the YouTube and Facebook channels regarding whether we have divers on board to make repairs or retrieve equipment. So that's a very good question. We definitely have some divers on board. Mostly, I definitely know that a lot of the science party, we do have diving certificates. Myself, I'm a very passionate diver, but I would say that usually this type of operations do not involve and do not require the necessity of diving and repairing the things, because usually we have the ability with the Falcor 2 vessel of retrieving everything we put on the ocean to retrieve it back on board. So if there is something that needs to be repaired, that happens once it is in the hangar. So very good question. Thanks to all the people who's tuning in, and please keep leaving your comments and questions as we finish our sampling of a beautiful sponge. Earlier on, we saw pretty amazing creatures. I would say that one of the most surprising sightings was a Pacific white skate, very, very large one, approximately between 1 meter and 1.5 meters. So if you didn't see that, I would totally recommend that you go back in this live stream and take a look at that, because that was pretty majestic. So thank you guys. That was impressive. Thanks. We will keep going in our way to the summit of the seamount. This is an ancient volcano. So part of what we're exploring here is not only the biological communities associated, but also the geology of this place that can tell us more about the history of this seamount and the geophysical processes involved. Tenemos una pregunta en el chat de YouTube, y un gran saludo para mis colegas costarricenses. Para mí es de verdad que un honor y un placer poder estar acá desde el buque de investigación Falcor 2 del Schmidt Ocean Institute. Nos hacen una pregunta de cómo ha sido la experiencia con los centros educativos y si hay otras actividades planeadas. Así que muchísimas gracias por esta pregunta y comentario, ya que es importantísimo dejar claro que las expediciones del Schmidt Ocean Institute y de nuestro equipo a bordo no solamente es recolectar evidencia científica, geológica, biológica, microbiológica, geofísica, sino que también poder trasladar esto a la población en general. Okay. Okay.
2 or 3 minutes.
Okay, got it. So we will catch up with the ship. We will remain steady for a little while. Y les comentaba que de los objetivos principales de estas expediciones es el componente de comunicación, educación e impacto en la sociedad civil y también hacia las autoridades gubernamentales. Hemos estado teniendo bastantes actividades asociadas a centros educativos, no solamente en Costa Rica, sino que también en otras partes del mundo. Esto usualmente son actividades que se programan con antelación y se hacen lo que llamamos ship to shore, o transmisiones en vivo desde el barco, en donde no solamente tenemos la posibilidad de comentarles a las personas con las que hacemos la conexión, les contamos un poco de la ciencia, pero también les hacemos un tour en vivo por el barco para que ellos puedan ver de primera mano cómo es la dinámica de investigación y de procesamiento. Sí. Can I do a zoom? Estamos esperando el barco. Mientras vemos este bonito espécimen de un octocoral con... Sí, quiero un ok. ¿Y ese qué es? ¿Un bambú? Pero parece. Sí. Acá vemos, y tengo el honor de estar acompañado aquí en el cuarto de control por la experta en octocorales, la doctora Odalisca Briti de la Universidad de Costa Rica.
Change my thrusters a little bit, so we'll try to stop that shaking.
Okay. Ay, la verdad es que yo creo que ven nos vamos a estar, pero estamos haciendo. Sí, sí, ok. Dino, eso está mal, ¿no? La cuestión, entonces, una ramita.
Sí, hagamos una ramita que tenga ramificaciones.
Okay. So I think we will go and try to take a sample of this one as well.
But first and close up. Close up, and
Sí.
then we can put
Mhm.
like
Can we turn on the lasers, please? Miguel, are you okay with it? Yeah.
Mira, es compartida. Esas esponjas no las agarrado, ¿verdad?
No, no, nada más que decir. Sí.
So, this is the base. What's on the?
In a second there.
I'm trying to get a good spot on the top right one for you.
Yeah. So what we're seeing on screen might likely to be associated with bamboo corals. We can see also
There we go. That's what we
a
do.
type of echinoderm, type of brittle star associated as a host. This is pretty common to see at these depths. So we usually say that corals are the luxury hotels of the deep. And well, you have the perfect example here. Estamos viendo en pantalla un octocoral, probablemente un octocoral bambú que se le conoce, con un ofiuroideo asociado. Y también aprovecho para terminar de comentarles sobre las actividades que se tienen de comunicación y educación asociadas. Usualmente todo está planificado meses atrás, ya que hemos recibido muchísimas solicitudes de conectarnos y poder llevar este mensaje a escuelas. Como les comento, a pesar de que nos gustaría conectarnos con todos y todas, esto lleva una planificación. Así que si ustedes que nos están conectando desde diferentes partes del mundo, Costa Rica, si tienen alguna idea o alguna solicitud, propuesta para hacer estas conexiones con centros educativos, estamos muy interesados y tal vez no se pueda en esta ocasión, pero sí que definitivamente les invitamos a que se pongan en contacto con algunos de nosotros y tal vez se puedan coordinar para el mes de diciembre que vamos a estar regresando y en nuestra segunda parte de esta expedición. Nosotros también vamos a estar haciendo esfuerzos de transmitir todo esto que hemos estado viendo, no solamente durante el tiempo de la expedición, sino que también después de, a través de exposiciones de arte, a través de eventos, charlas, artículos en medios periodísticos. Así que, pues de verdad que los invito y les invito a que si tienen alguna propuesta para centros educativos, por favor nos lo dejen saber, porque para nosotros es importantísimo. Diría que de las cosas más importantes para este tipo de expediciones, para tener un impacto verdadero y empoderar a las nuevas generaciones de todo este patrimonio y estas maravillas que vemos en las aguas profundas de nuestro país.
I want you in their arm, around their arm. You and your green light.
So we will try and take a small sample of this coral that we see on screen that has an ophurate associated. This is a very common association in the deep. So we have experts on board that specialize in corals and octocorals, and they will be taking a closer look at these specimens that we can sample. Please keep leaving your comments and your questions in our YouTube and Facebook channels. We see very, very interesting questions. So there are some questions in regards to the oxygen measurements. So we have two main, or what you see on screen, it's two main oxygen let's say, measurements. So the first one that you see is the oxygen dissolved in the water, and the units are micromolar. So that's the one that's currently reading 96.56. That's micromolar. This is a common unit for scientific research, but you might also find this in milligrams per liter or milliliters per liter. This is very important for us to assess the hydrographic and oceanographic features and characteristics around these seamounts.
What would you like?
Well,
Yeah. Maybe.
and below this oxygen measurement, as we see the sampling of this beautiful coral specimen, we also see one that says O2 sat, and that's the oxygen saturation. So for you to have an idea, the oxygen saturation at the surface is usually about 100%. So as we go down, that's the capacity of the water, or a relative measurement of the oxygen that water can hold. So this measurement, in conjunction with the temperature and salinity readings, allow us to better assess the hydrography and the oceanographic features around these seamounts. It's actually very interesting to see that actually the oxygen is kind of low here in comparison to other places that we've been visiting in previous dives. But again, we have these sensors on board the ROV SuBastian. So while we do, and this is also part of my job here, as we sample this octocoral that you can see on the screen, part of my job is taking a closer look at this data and create distribution maps of these different parameters, such as oxygen, salinity, temperature, because this can tell us more about the water column features associated with this seamount. So thank you everyone for these great questions, and we hope you're enjoying this live stream as much as I do. And this is kind of a hard one. Can we use the other arm? Or would you just like to avoid to have the same thing as the sponge?
Yeah, we can give it a shot. We're gonna try to grasp it.
Yeah, try to wiggle a bit, but yeah, if not. So as we see this sampling, and we were previously talking about oxygen concentrations, I would just also take advantage of the moment, because part of what we do here in this expedition is not only assessing and evaluating the seafloor, but also what happens in the upper water column. So this is part of my contribution in this expedition, and part of the instruments that we have on board let us know and give us the possibility to assess and have full water column profiles of different parameters that might tell us a bit more about the distribution of these oceanographic parameters, and also the circulation in this area of the Eastern Tropical Pacific. There is a really important feature that's called the oxygen minimum zone. And this is a mid-water feature that is usually located between 200 and 800 meters depth. And this is a naturally occurring oxygen depleted zone in which the oxygen concentrations drop almost even, sometimes to to anoxic concentrations. That means zero oxygen. And this is mainly associated or attributed to the microbial activity that happens because of all the sinking particles that are usually produced in the upper water column at the surface, that we also know them or know this all these particles usually as primary productivity. So you can, for example, take examples of algae, different crustaceans, etc., that what we call plankton and phytoplankton. And some of these organisms end up dying and they sink down to the bottom of the ocean. So while they travel to the bottom, they get, let's say, some microbial interactions. And at this particular depths, between 200 and 800 meters in this region of the world, all this microbial activity ends up exhausting the oxygen. So this oxygen minimum zone is really important to understand the distribution of many important species, usually also associated with fisheries, because as you can imagine, animals usually look for water that are that have enough concentration of oxygen. So this will depend on the life form, and this is what we're trying to understand here, and also in other expeditions that have been happening recently. But also a really important part of assessing these oxygen minimum zones is because as we experience the effects of climate change, there is several models that predict that this oxygen minimum zone can expand their thickness. So this could have an impact on the distribution of the biomass and species in the ocean overall. So again, thanks for your questions, and keep leaving your comments. We are reading them as we sample this octocoral. So apparently people is really interested into oxygen, so I will take advantage of the moment. And just before I hand it over to another good colleague of mine, I will just finish it up saying that because we have a question that why we do have oxygen below this oxygen minimum zone, and it is because of the currents, of the effect of the currents and what we call ventilation. So at those particular depths, you have the microbial activity exhausting the oxygen, but also you have, let's say, not as much as ventilation as the ventilation you have deep in the waters. And this is because deep waters usually sink, and because of their of the water mass characteristics, they can hold a lot of oxygen. So that's the main reason why we find this oxygen minimum zone limited to the first 1,000 meters, but deep down and down to the deepest trenches, below 1,000 meters, you usually get back to the normal oxygen levels. So yeah, very good question. And for people interested, I invite you to Google over because this is a pretty particular topic that has been recently, let's say, relatively starting to be studied and better understood. So for me, it was a pleasure. My name is Sergio Cambronero, and I will hand it over to a very good colleague and friend of mine, Doctora Celeste Sanchez from Universidad de Costa Rica, who will be joining you in this great dive that we are currently having at Tengosed Seamount. Muchísimas gracias a todos. Los dejo con la doctora Celeste Sánchez del CIMAR de la Universidad de Costa Rica, que los va a estar acompañando durante este buceo que estamos teniendo en el monte submarino Tengosed. Pura vida. I think we just advanced like 200 meters. Yeah, I don't. Nada, está ahí como. No, no, no.
So again, as we do that handover, scientists are going to kind of just catch each other up. Sergio's going to go over with Celeste what we have been collecting and been looking at. And we also have a little pilot handover going on here. Sitting down in the pilot seat, we now have Adam, and co-pilot's going to be Scott. And I'm still Alex. That's it. I'm currently in as the data logger from SOI, but I serve as the multimedia technician on board for this expedition.
Hi everybody, I'm Celeste Sanchez Noguera from the University of Costa Rica. I will be here with you for the next hour. Thanks for staying tuned and for joining us in this Octopus Odyssey. Hola a todos, bienvenidos a esta nueva hora de transmisión. Yo voy a estar con ustedes por la siguiente hora. Mi nombre es Celeste Sánchez Noguera, soy investigadora de la Universidad de Costa Rica en el Centro de Investigación en Ciencias del Mar y Limnología y también docente de la Escuela de Biología de la Universidad de Costa Rica. Muchísimas gracias por estar acá conectados con nosotros, por acompañarnos. Realmente es un placer poder abrirles esta ventana a las profundidades de nuestro gran gigante azul y pueden dejarnos sus comentarios y sus preguntas en el chat. So we are just now in waypoint 4 and continue our exploration. We are on our way to waypoint 5. As you can see here, there are really beautiful sponges. They have like this bell shape. Actually, they resemble a lot of one flower that is very common in Costa Rica called Queen of the Night. So yeah, this is really beautiful. And on the bottom, we can also spot these little white spots. They are actually polychaete tubes. And these worms included in their tubes some sediments. That's also why they look with this particular texture. If you have any comments or questions, please just send it in the chat, whether via Facebook or YouTube. We will be really happy to answer any questions regarding the dive or regarding other information you want to know about the whole expedition. Can we take a close-up look to the sea cucumber and also to the sponge? I think it is a sponge, the one that we have on the upper left, please. Over the last couple of dives, we have been spotting a lot of sea cucumbers. This is just one of examples of these specimens. We are going to have now a closer look to take pictures. We are not sampling all these specimens, but we are for sure documenting everything with pictures and videos. So in order to do that, we take like broader pictures. We include scales with the laser that we have on the SuBastian. ROV, and we also take close-ups of all the animals, and this will help in the future our colleagues and other experts to try to identify the organisms even if we haven't take or collect a sample. Bueno, me avisa cuando tiene las tomas para alejarlo. Actually, I would say this looks a little bit different from the ones that we have been having before, especially because of these whitish spots. Yeah, okay. Can we take one with the laser, guys, please? Thank you. So there are the laser lights. Just as a reminder, the distance between the lights is 10 centimeters. And these help us to actually have a roughly estimation of the measurement of each animal. Okay, can we move to the sponge, please? Thank you. There is a crinoid on the rocks, and that over there, it looks like a sponge. Yeah, it's definitely a sponge. Yeah, okay, thank you. And now we are going for the close-up. Actually, as you can tell, there are some animals, some crustaceans also like crawling on the sponge. Okay, yeah, great. Thanks a lot, guys. So here with me in the control room, we have another Costa Rican colleague, Gwendolyn Matamoros, also from the University of Costa Rica, one of the experts on octocoral, so Dalis Cabridi, and just behind the wheel of SuBastian are our pilots, Adam and Scott. We have Alex as one of the marine technicians. There we can see another of these sea cucumbers, one of the transparent ones. And more of these beautiful sea sponges. En este buceo en particular hemos podido observar varias de estas esponjas que para aquellas personas que viven en Costa Rica o todos los ticos que se encuentran fuera de Costa Rica, a mí me recuerdan particularmente mucho a la flor reina de la noche por la forma que tienen. Son bastante lindas realmente. Acá estamos viendo en esta toma un octocoral del género Antomastus. Y lo que se ve en las rocas o en los basaltos, en estas formaciones de lava, son unas manchitas blancas que son en realidad tubos de poliquetos. Los poliquetos son gusanos marinos y en este caso los poliquetos construyen tubos y lo que hacen es incorporar granos de sedimento a los tubos. Entonces, por eso es que se ve que tienen como una textura diferente y un color también diferente. Muchos de ellos, inclusive, el tubo lo hacen a partir de carbonato de calcio. Guys, can we have like a look at this sea cucumber, please? So, this is another species. And as you can see, it's just really like a paradise for echinoderms. This is just like the third different sea cucumber that we spotted in less than, 5 minutes or something like that. So we're going to do exactly the same. Take like a general picture with the lasers on and then a close-up look. Estos montes que hemos explorado realmente están llenos de una gran diversidad de organismos y en términos de equinodermos y de pepinos de mar, ha sido increíble ver la gran cantidad de especies diferentes que hemos encontrado. Este es, de hecho, el tercer pepino de mar diferente que vemos desde que comencé mi turno hace menos de 5 minutos. Entonces, para nosotros es muy importante documentar todo esto lo mejor posible. Si bien es cierto, se han recolectado especies o individuos de algunas de las especies que hemos visto, no estamos recolectando todos los organismos. Entonces, lo que hacemos para posteriormente poder realizar identificaciones de los que no se realizaron recolectas es tomar fotografías. Se toman fotografías amplias utilizando una escala con las luces láser y además de eso se toman acercamientos de los organismos que en el futuro le puedan servir a los expertos para poder realizar la identificación. Acá lo que estamos viendo ahora es una estrella quebradiza, también conocidos como ofiúridos. También pertenecen al mismo grupo taxonómico de los pepinos de mar. Ambos son equinodermos. So we are just doing now the close-up of this sea cucumber, and you just had a glimpse of a brittle star very close to the sea cucumber. Remember, the brittle stars and the sea cucumbers are within the same taxonomic group. They are both echinoderms. So we continue now our exploration of this seamount Tengosed, which in Spanish literally means I'm thirsty. And the reason why they name it this way is because this is thought to be one of the recharge sites in this area. So it's basically an area where cold water it's being taken, or is being there is like an intake of cold water that then travels through the cortex, and eventually it will be discharged in other locations. Apparently, Dorado is one of these locations where the discharge occurs with a little bit higher temperature. So this is one of the main goals of this expedition: recharge and discharge. Now there is a little bit of change in terms of the geological scape. As you can see here, the rocks are a little bit more like break into smaller pieces. They are not so consolidated into big ones. And this also like one of the important features of this kind of seamounts. Actually, I'm going to ask, because we have an expert on board, a geologist. I'm going to ask her if she wants to sample one of these rocks. Guys, can we zoom in? Thank you. Oh, what? Look at that. Well, that's a shrimp for sure. But actually, we have a sponge, a little sponge as well. But I think on the upper left, we spotted something different. That seems, wow. Esto que está aquí a la izquierda. Can we just take a close-up of this one, please? So we can take a picture and also with the lasers. Thanks. So we are not really sure this could be a sponge or something else. So we are just taking pictures for later analysis. Okay, thanks. Yeah, something like that. Yeah, thanks, Adam. No, we are going to continue. Yeah, thanks. Yeah. Yeah, I think we can try to speed it up a little bit. So we try to make it to waypoint 5. Unless we see something really astonishing. Yeah. ¿Sí es una esponja?
Hello, on the bridge. We got an updated waypoint for you. Thank you, waypoint. Thank you. Thank you.
So we're going to try to speed up a little bit so we can make it to waypoint 5. We still have a long list of waypoints to explore today. So our goal is to try to reach the summit of this seamount. On the way, as you can tell, we have more of these beautiful sea sponges on this rubble field. These are all basically rocks that fell down from somewhere else. More polychaete tubes, some other sponges on the back. There is one octocoral sitting next to a sponge. Those long shade sponges that we are just seeing are also called Venus's baskets, and they have like a really beautiful story because usually when they are forming when they are already formed, some crustaceans, some small crabs get into the sponge when they are larvae, and then they just start to grow in there. So at certain point, they are just too big to actually being able to escape from the sponge. So usually we can find like a female and one male of these shrimps.
So this stuff up above, this would be intact pillows that we could try to sample if you can see something that might be breakable.
Okay.
Not the rubbly stuff, but
Yeah.
the stuff that's all coherent
Okay.
is intact.
Okay.
What are you looking at?
Straight at that, just kind of just below the rocky spot.
It's a dead sponge. Yeah.
It's sea lots of these
No, some kind of polycate casing.
Yeah, those are polycate, yes. So you mean that if we find something that we can just retrieve from this area?
Yeah, something, I mean, it's obviously attached, so
Yeah.
it's not going to be easy for the pilots.
Yeah.
But yeah, off to our left that we were passing, also anything in this big pile would be great. That looks new.
Yeah.
I think they sampled that earlier.
Yeah, I think so. Yeah, that seems like this.
Scientists are conferring about possibly collecting from this area.
So we are exploring this part just to see whether we are able to sample one of these rocks. We have here in the control room with us Dr. Beth Orkut. She is one of the co-chief scientists of the expedition. And we are also very interested in sampling these rocks because it can tell us a lot about the geology and the formation of this seamount. Estamos en este momento tratando de ver si es posible recolectar alguna muestra de esta zona rocosa que estamos viendo en este momento, porque este tipo de formación nos puede decir bastante información en términos geológicos sobre el origen de este monte submarino que estamos explorando en este momento. Tengo fuente. Nos acompaña en el cuarto de control la doctora Beth Orkut, quien es co-líder científica de esta expedición.
Yeah, there's like a crevice in here,
There
but
one
there's
there?
also one down at the bottom of the screen just below that crab that looks like you could maybe get.
What about that inside the, yeah, inside there, that,
Like this one?
Yeah, that one would work, Scott, or down below in the lower left where the crab is. Or not crab, spot lobster.
which piece is it?
The one with the anemone on it.
That's pretty big.
Is that
Yeah,
big?
that's very big.
Can we get the scaling lasers on?
You're thinking like crack that chunk off.
Well, they are.
Yeah, there they are. I see them. So that's probably, yeah, 25 centimeters. That's probably too big.
Mhm.
Come on.
This one up here might be all right.
Should we try to go for that one?
That looks like it might be separate.
That's small. Well, we'll see what we get. Let's try it.
Okay, let's do some rock sampling then. We are going to try to
Can I get
collect
some zooms before you do that?
that rock.
Yep. Oh, I didn't realize you were using it to maneuver.
Vamos a tratar de recolectar un fragmento de roca de este monte en el que nos encontramos. Como pueden ver, la mayoría está bastante consolidada, así que tenemos que aprovechar la oportunidad que estamos viendo de un fragmento que pareciera ser estar levemente suelto. Vamos a ver si lo logramos y si no, entonces tenemos que continuar explorando y buscando otros fragmentos que podamos recolectar. Como les comentaba hace un momento, para nosotros es bastante importante también recolectar rocas, además de organismos y muestras de agua, porque las rocas, por un lado, nos permiten estudiar cuáles son los microorganismos que están viviendo en este tipo de ambientes, pero también nos permiten hacer análisis geológicos y determinar cuál es o cuál ha sido el proceso de formación geológico de este tipo de montes submarinos.
Get some screen grabs, please, Wendy.
laser on it.
Kind of a big potato back in there.
So we're attempting to collect this little fragment of rock that is just there.
Imagine yourself as a chisel.
Let's see if we are lucky enough.
Okay, so let's, yeah, this will go in a bot box?
This is going to a rock
Yeah,
box,
we're
right?
going to buy the buy box one, upper left, upper port side.
Just in case it's friable.
Yeah.
You want it here or in the rock box?
No, I want for my, what is that?
Antomastus, that's an octopus.
Beth, are you ready?
Hold on one second. Yeah, these were supposed to be reserved for rock samples, not for
Yeah,
biology.
I didn't put it there.
It's
Yeah.
okay. Yeah, so it needs to go in A.
Okay, so it has to go in bio box 1A.
I don't think
Yeah,
they
please,
get it
yeah.
out. Ah, okay. So they're gonna try it as file, okay. Yes, go for it.
Okay.
This is exciting. It's like a very heavy
The
duty
ledge that's
work.
right behind you, you think that's grabbable? This thing, right behind the nips.
That's okay. Yeah, it's at a bad angle. Let's just pump around and see if we can find something really.
Yep.
Just reaching your hand into deep dark holes in the deep sea. Nothing could be in there.
The port side, when we were first coming around this, it looked like there might have been some things that were grabbable, but whatever you see.
I was gonna say it's rubbly here, but I don't know if that's too far gone from what they're wanting.
Yeah, the thing is, we don't want those rubble rocks because they fell from somewhere else, and apparently they want to have like the real rock. So
Might be able to put my ball around that right there.
maybe this one
On
here.
either side of this, 1, 2, or on either
It's
side.
probably too big.
Wiggle it off.
Yeah, it is.
I don't think so, compared to the sponge.
¿Y esto? No.
No, porque ese
Okay.
viene como quebrado.
Okay.
Tenemos que quebrarlo.
Okay.
We have to break.
Yeah, we have to actually break it. We cannot grab any of the
For me,
pieces.
we can take whatever, but for her, we need
to break it.
something that is
Yeah,
outcropping.
yeah.
What do you think it's at? We'll just do a test. Yeah. And then we'll take pictures. I think that's fine.
So as you can tell, it's really not an easy task trying to figure out what is the best place to just crack the rock. Mm-hmm.
Stable yet. I'm calling.
Okay, you want to take pictures first?
Yeah, can we get a still, please, Andy? Actually, the rock, well, we can try these, but there's one just above this shot. There.
Ooh, yeah.
It looks like a-
Okay.
See big rock? Little softball sized rock, just on top of it.
Crack it.
Oh, esta.
Oh. This guy?
Yeah, it's not bad.
Okay.
I can bump up.
I might be able to. It's gonna be pretty high. Having to reach.
Yeah, go for it.
That's fine. Sorry, Splash.
Might be a sacrifice on that.
Okay, and here we go. We are trying to get for that softball shaped rock.
Nice size. Rock sized.
Ooh, it's apparently too big. Seguimos en estos momentos en la misión de tratar de recolectar un fragmento de esta roca. Sin embargo, no es una labor sencilla. Tenemos que encontrar una zona donde se pueda fragmentar fácilmente la roca. Y como pueden ver, en este momento estamos intentando con un pequeño fragmento que al parecer es levemente más grande de lo que puede abrir la pinza de SuBastian. Vamos a continuar avanzando un poquito más para ver si encontramos tal vez algún borde que sea más fácil de quebrar o de recolectar. So we are going to continue exploring the surroundings of this area to check whether we find like an edge or something that is a little bit more easy to break or to grip with SuBastian's arm.
Talking this one here.
I can reach from the back and front and move it back and forth. I mean, it looks like it's
Yeah. I mean,
slender enough where I can get both.
It's just amazing to hear and to see the pilots of SuBastian, like how they have to think like all the possibilities and how to approach this kind of sampling, because they have to find the best way. They have to use, for example, stabilizers to keep the rope steady.
The one on the left would be preferred because it's still got some of the rind on it in addition to the ground mass. Is that feasible for you, Scott?
Yep.
Take some screen grabs, please, Wendy. And can we get the scaling lasers on, please?
They're just out of view.
There they
Yeah,
are.
there.
Okay, and another screen grab, please, Wendy. Okay, let's go for it.
So in the same way as we do with all our biological samples, we also have to take pictures with scale of our geological samples, just as a reference. De la misma manera que hacemos con las muestras biológicas, también tenemos que tomar fotografías con escala cuando vamos a recolectar muestras geológicas. Todo esto les recordamos que nos ayuda en el caso en el que luego se tengan que hacer reconstrucciones. That's great. You just did it. Okay. So, okay, let's try to get that rock into the bio box before it breaks apart. That's really nice.
Screen grabs there, please, Wendy.
Lo logramos, muestrar o recolectar un fragmento de esta roca. Estamos en este momento rotándola para tomar fotografías desde diferentes ángulos y vamos a colocarla en uno de los contenedores que tenemos para muestras.
It needs to go on the A side.
For sure.
Okay, that's nice.
That's the new book from the author of Goodnight Moon. Thank you, Rock. Thank you, Scott. Thank you, giant
Before we fly
robot.
away, can we get a zoom in on where the rock broke?
Okay, so now we're getting a zoom in.
Look at that nice line.
Yeah.
It's definitely a lava. It's hard to tell from here when we get on the ship to see if that's actually an alteration rind or if that's just stuff that's precipitated on the rocks over time.
So this piece of rock will go through a lot of scrutiny back on shore, and that will tell us a lot of information in terms of the geological formation of this seamount, Tengosed. Esta roca que acabamos de recolectar luego va a pasar por diferentes análisis de vuelta en tierra y nos va a permitir conocer un poco más sobre el origen geológico de este monte submarino Tengosed. So this was actually one of the goals of this dive, trying to sample some rocks from Tengosed Seamount. So are we trying to get another piece?
Yeah.
Awesome.
That way we have one for microbiology.
Okay, and
geology.
can we put it on the same box, right? Yeah. Okay, so then we are going to try to get another piece of rock. In that way, we can just split it. One will go to the macrobiology group, and the other one will go to the geology group. Vamos a tratar de recolectar otro fragmento de esta misma roca. Así podemos dividirlo y enviaríamos uno de los fragmentos al grupo que trabaja con microbiología y el otro entonces podría pasar a mano de los geólogos para continuar haciendo más estudios.
That's rubbly.
So small it fits through.
Oh.
You want that?
Yes, we'll take it.
Yeah, right.
It's volunteered itself.
So we are just taking whatever we can. This little piece just got stuck into Sebastian's arm. So we are going to try to put it into the bio box along with the other sample that we just recently collected. Este pequeño fragmento se quedó atascado en la pinza de Sebastian, entonces vamos a tratar de colocarlo igualmente en la caja, en la biocaja, el contenedor, junto a la otra roca que recientemente recolectamos. Y vamos a tratar igualmente de recolectar otro fragmento. So we are not going for more? We are not going for more?
Yeah, I think
No?
that's enough.
Okay, so our co-chief scientist just told us that that seems to be enough. So apparently we are not taking more rocks, and then we are just going to continue our way to waypoint 5. So do you want us to try to sample more rocks, or that should be enough for this
That's
dive?
enough for this diet for
Yeah,
now,
for
and then
this
if
dive,
we
yeah.
we'll see what we see at the
In
summit.
the summit,
So I
okay.
think right about now we're like halfway through the dive and
Yeah.
halfway through our waypoints.
Yeah.
So that's good,
Okay.
and we'll see if we see anything else different on the summit.
Okay, perfect. Thanks a lot, Beth. Okay. Adam, can we just go a little back and zoom in one of these small little branches?
Just point where?
It's on the left. I think it's just out of the frame right now. It was this one here, Wendy. Yeah, in that one, please. Yeah, thanks a lot.
You want to sample that or just do
¿Qué quieres? ¿Solo acercamiento? Yeah, it's just like to have like a close-up picture. Thanks a lot. Okay, there are the lasers on just for scale. We are going to try to take a close-up look at this, though there is a lot of resuspended sediment from the rock that we just recently broke. So let's see if it works.
that? It's just a lot of stuff.
Yeah.
It's kind of in a weird craft. You can use the camera if you like.
Yeah. Creo que eso es lo mejor que podemos hacer porque podemos hacer mucho sedimento, ¿está bien? Yeah, that's okay. Yeah, we can just keep moving. Thanks. There is another octocoral and a bunch of polykid tubes and sponges of different kinds of shapes. Continuamos nuestro camino hacia el punto 5. Estamos más o menos a la mitad entre el punto 4 y el punto 5. Y podemos ver acá igual nuevamente tubos de poliquetos, muchas esponjas de diferentes formas, tamaños. Vemos en la imagen también un pepino de mar, varias estrellas quebradizas. It is really impressive how the seascape changes from one side to the other. Like Tengo said, it's definitely different from, for example, Dorado, or even from the dives that we were doing previously in the unnamed mount. So we are right now about 2100 meters depth, and we are on our way to the summit. We still have at least 5 more points to actually get into the summit. So we will try to get there in a couple of hours. I really love those bell-shaped sponges. They definitely look like flowers. esas esponjas con forma de flor, como les decía antes, se parecen un poco a las flores comúnmente conocidas como reina de la noche, realmente son impresionantes. Y definitivamente el paisaje submarino es bastante diferente en todos los buceos que hemos visto. Eh, este de acá está dominado por muchísimos, por ejemplo, tubos de poliquetos que están adheridos a las rocas. Además, hay bastante cantidad de esponjas de este tipo con forma de campana. Recordemos que en los buceos anteriores hubo lugares donde la mayoría de las esponjas eran igualmente esponjas de vidrio, pero del tipo de esponjas que se conocen como canastas de Venus. Entonces es realmente muy interesante ver cómo, pese a que todos estos son sitios que se encuentran en profundidades relativamente similares, en realidad tienen una composición bastante diferente. En este momento nos encontramos aproximadamente a 2100 metros de profundidad y les recuerdo que este va a ser uno de los buceos más someros o menos profundos que vamos a estar haciendo el día de o en estos días. La idea es tratar de llegar a la cima o a la cumbre del monte submarino Tengosed en este buceo y estamos en este momento acercándonos al punto 5 y tenemos todavía, nos quedarían 4 puntos más para poder alcanzar la cima. Entonces esperaríamos ojalá estar llegando a la cima alrededor de un par de horas para completar esto antes de que terminemos nuestro buceo del día de hoy. Les recuerdo que en este momento estamos trabajando con periodos de buceo de 12 horas, lo cual significa que SuBastian entra al agua a las 5 de la mañana, perdón, en hora local, y se espera que esté de vuelta en la cubierta a las 5 de la tarde en hora local. So just a reminder that today's dive it's like a 12-hour dives, which means that basically SuBastian starts around 5 o'clock in local time in the morning, and then it's expected to be back on deck around 5 o'clock in the afternoon. So this is going to be one of the most shallowest dives that we are going to be doing here during this Octopus Odyssey Expedition. And we are attempting to reach the summit of this Tengosed Seamount in today's dive.
Great. I think we're good to resume our move.
There is also like a lot of
Just happy to continue on to the waypoint, please. Very good, will do. Thank you.
a lot of close communication between the control room of SuBastian and the bridge, because the ship actually has to move along with the submarine, so they have to be always communicating to see whether they can move or they have to wait one shot we shot other. Es bastante interesante porque en este tipo de transectos que estamos realizando, el barco se tiene que ir desplazando en conjunto con el submarino. Entonces, tienen que estar en una comunicación bastante cercana para indicarse si pueden desplazarse o si tienen que esperarse en algún momento mutuamente, ya sea porque se están recolectando muestras o porque el barco, digamos, tal vez esté teniendo alguna situación en superficie y necesite que también el submarino tenga que retrasar un poco su desplazamiento. Pero en este momento estamos bien, estamos de hecho alcanzando ya lo que sería el punto 5 y nos quedan todavía 4 puntos más para llegar a lo que sería la cima del monte submarino Tengosed. So we are just right now reaching the waypoint 5. Están súper lindas. That is actually quite a beautiful geological or rock formation, the one that you can see on the lower right corner. Bueno, por lo menos llegamos al punto 5.
¿De cuántos?
Faltan 4 más, son 9 en total.
Estamos por la mitad del camino.
So there is another Antomastus, one of these beautiful octocorals. They seem like flowers, actually, the same as the sea sponges that we have been looking all over the dive. Yeah, so we are on waypoint 5 right now. This has been a very interesting dive. We spent quite a lot of time trying to collect one of the rocks, to sample one rock that it was actually very important for this dive, in order for us to try to understand what is the geological formation of this seamount, Tengosed. So we can see there in the frame, there are more of these beautiful sponges. There is one fish over there just passing by. Actually, I think this is the first fish that we see in this hour dive. Thank you. A lot of octocorals, big, small ones. And again, a lot of these polychaete tubes. Muchísimos de estos tubos de poliquetos vemos en algunas secciones de los basaltos, de las lavas, de las formaciones de lava. Recordemos que estos son gusanos marinos que producen o forman su propio tubo donde ellos viven, y muchas veces estos tubos están formados en parte por carbonato de calcio, pero también ellos incorporan sedimentos o granos de sedimentos en el tubo. Entonces eso les da este color y textura tan particular. It's just like a beautiful garden of bell-shaped sponges. It's just amazing. I really love this kind of sponges. And I think this is actually the first place that we have been diving where there's such a big quantity of these sponges, unlike the other places where the Venus's basket sponges were the most common ones. the sponges are very fragile organisms, so we cannot sample all the sponges that we are actually seeing, but then we just take like very good pictures of it. That's an octocoral with a brittle star. Usually there are like very close associations between octocorals and brittle stars. Usualmente hay bastantes asociaciones cercanas entre los octocorales o corales blandos y las estrellas quebradizas. Entonces, no es poco común observar que hayan estrellas quebradizas adheridas a octocorales. Nuevamente acá vemos un octocoral del género Antomastus que me recuerda bastante a un diente de león. La forma que tiene es bastante particular y bastante linda, bastante agradable a la vista también. So that's like a beautiful shot of this Antomastus octocoral. And just in the back, there is one sponge, another octocoral. Actually, love a lot of different encrusting sponges and some different octocorals. That one over there actually looks like a small black coral. So I'm close to finish my shift now, and I will be handling over to my dear colleague and friend, Dr. Jorge Cortez from University of Costa Rica. He's just such an amazing scientist. He's one of the also experts in coral reefs in the region. So it's also one of the co-chief scientists of this expedition. So thanks a lot for staying tuned with us. It's really a pleasure to have you all guys here and also to read your comments. Muchísimas gracias por acompañarnos, por mantenerse en sintonía con nosotros. Realmente es un placer que nos acompañen en esta exploración de las profundidades del océano costarricense. Voy ahora a darle el paso a mi colega y estimado amigo, el doctor Jorge Cortés. Él es realmente una eminencia a nivel nacional y también a nivel internacional, un experto en corales y que se ha dedicado mucho a la exploración de zonas profundas de nuestro país. Entonces, me despido de ustedes y los dejo en compañía de don Jorge.
Buenas tardes, buenas noches, buenos días. Les habla Jorge Cortés de la Universidad de Costa Rica. Yo voy a estar con ustedes durante la próxima hora explorando este impresionante monte submarino llamado Tengosed. Hello, my name is Jorge Cortés from University of Costa Rica, and I'll be with you for the next hour exploring this interesting seamount called Tengosed, and here it's in this crab with 2 parasites on the antenna. In the background, the little light blue circles, those are sponges. It's amazing how many of these sponges we see here, these bell-shaped sponges. We haven't seen that many in a single place. Se llama mucho la atención estas esponjas con forma de campanas. No habíamos visto tantísimas juntas. De que seguimos navegando hacia la parte más alta de este montículo, de este monte submarino. Vemos algunas de las paredes son bastante verticales y todo lleno de vida. And we keep cruising up these walls to the summit of this seamount, and just every rock is covered with life, from large animals that we can see here to very, very small animals. We have a sea star right on the center, surrounded by sponges, small sponges. And those tubes you see, those are polychaetes. Here again, some more octocorals, the antomastas, that one, the feathery polyps. Creo que llevamos uno de estos. Yeah, here we see on the right is a glass sponge called Venus basket, I think it was the name in English. Aquí vemos más antomastes, octocorales, unas esponjas de vidrio, las campanas estas que hemos estado viendo, y esas manchas blancas que vemos sobre las rocas, pensamos que son, algunos de esos son poliquetos, gusanos marinos. Y vemos corales negros. Oh, yeah. Focusing into one of these sponges. They're all glass sponges, as opposed to sponges that have carbonates. These ones are siliceous, and here we see another antomastes, the octocoral, black corals. Let's see this here. This one. It's a sea pen. Eso que vemos en el centro del cuadro, eso es una pluma de mar. Les hemos visto varias al lado del antomastes. Vemos corales negros, esponjas, los gusanos. Es una gran diversidad de organismos. Here I see this on the center is the sea pen right next to that big antomastes, that octocoral. That sea pen is also an octocoral. I also see some polychaetes and worms, more sponges, black corals. This is the expedition called Octopus Odyssey because that's one of our objectives, to check out some octopus, and which we found on our first couple dives, and we're still looking for more. And but meanwhile, we're seeing all this impressive creature and collecting a good number. They will be studied by scientists from Costa Rica, from many other different countries, and they will all be deposited at the Zoology Museum, University of Costa Rica, and will be available for study by scientists from anywhere. Sí, esta expedición se llama la Odisea de los Pulpos, porque nuestro primer objetivo era encontrar un sitio donde habían visto pulpos hace una década y los encontramos en los primeros días. Se pudieron colectar, eran necesarios para poder estudiarlos, y ahora seguimos buscando. No hemos visto en este sitio, pero mientras tanto vamos viendo todos estos otros organismos. Hemos recolectado una buena cantidad que van al Museo de Zoología de la Universidad de Costa Rica. Ahí van a ser estudiados por científicos locales y extranjeros con los que colaboramos. With all these studies that we do, we prepare scientific publications that is, go to journals and go for peer review and eventually publish. But also all that information is used to advise government about importance of these deep areas for protection and conservation. La información que generamos con todas las observaciones y colectas, eso va para publicaciones científicas en revistas indexadas con revisión por pares, pero también la información la pasamos a lenguajes más sencillos, más aplicables para el manejo de estas zonas, y eso se lo pasamos a las autoridades nacionales para tratar de proteger y conservar más estos ambientes. Let's take a look at some of this very, very thin wiring. We're going to take a close look at one. So pareciera, ¿verdad? Yeah, this small wiry organism is probably a black coral. Sitting next to me is Wendy Matamoros. She's a student from University of Costa Rica studying this group of black corals. Yeah, no doubt it is. Lo que estamos viendo es un coral negro. Aquí conmigo está Wendy Matamoros, estudiante del posgrado en la Universidad de Costa Rica, que se está especializando en este grupo de organismos. Esta es una buena oportunidad para ella para observar. There's another, this one over here. Aquí estamos viendo otros, estos son ya organismos muy pequeñitos. que se me parece un poco a un grupo de hidroides calcáreos del grupo de los estilastéridos. Yeah, that white one, small white one in the middle, looks like a calcareous hydroid of the group of the stylastrids. A bivalve. Can we like with the suction trying to get one of these guys?
The suction? I don't know. Will it pull off the rock really easy? Is that what you're thinking?
We're not sure. We've never collected this one before.
The last, we tried the other day to take one of these corals with the suctioner, and it just gets stuck in the hose.
Okay, yeah,
But we
that
can
could.
try to collect and put in a coral cover.
Estaban dando unas tags para que agarres ese otro delgado.
¿Cuál?
Ese delgadito.
El delgadito.
Sí.
Okay, then we'll use the last quiver for that. Let's get when like that one that looks relatively large. Pick it up from the base.
El delgadito. ¿Ya lo vieron de cerca? Ok, entonces lo vieron. Al altero, ¿no?
Ya no mandó órdenes.
Ya le mandó la identificación. Ay, sí, mira.
Sí, velo.
Ah, mira, qué chido.
Belleza. Sí.
Pero no muy grande.
Could we see the laser to have an idea? Okay, small. It's like 10 centimeters across. It's good.
Okay.
Great. Yeah, so we can take it from the base. Would be excellent.
Try to get the entire thing for it.
Yeah, entire. Yeah, from it's lower on the base as lower you can go. Yeah. Actually, yeah, like in this image, like seems to be right there where it's attached.
So we want to go for the 8.
Yeah, let's see. Solo queda el 8, ¿verdad?
Eso era un braquiópodo.
No, me parece que es un bivalvo. Un bivalvo es Venus, pues que le dicen. Y es que voy a ver, es que no, porque ya no tenemos campo. No, tiempo, no, campo. Esos son los squeezes, esos son las inyecciones esas. Mm-hmm. Mm-hmm. We're collecting this black coral. You could see in proportion to the arms is a very small specimen.
Pushing the subway. Can't go anywhere else.
Estamos tratando de recolectar este coral negro que no habíamos visto y aquí vemos que es muy abundante. Vamos a ver qué obtenemos. Ah, good. Nice, nice. Está aquí el Wendy, de las estudiante que trabaja este grupo, muy contenta con un espécimen nuevo. With me here, as I mentioned, is Wendy, that she's studying this group of black corals. She studied the black corals of this country, so this is a superb sample from 2,045 meters depth. Here we see a good view of the specimen. Great. Mm-hmm. And water temperature out there is 2.17 degrees Celsius. And I remind our viewers that this place is pitch black, and we can see it because we have powerful lights on the ROV. Estamos recolectando este coral negro. Bien. Good, good. It's going in. Beautiful, beautiful. Excellent. Very good. It's a great specimen we collected there.
All right. That bivalve under the ledge looks like in the genus Ascent.
Uh-huh.
So it's the first one I've seen.
Yeah. Let me see if we could.
It's
Let me,
probably too big for the suction sample,
yeah,
which
that's
hasn't
that's
been used at all.
that's what I suspect. And if these boxes, those are for rocks.
Any heavy, larger items that won't go through the cracks?
Well, so we were thinking that bivalve that was next to the black coral.
Under the ledge.
Under the ledge there was a bivalve.
Yeah.
So let's go by there, take a look at it. Yeah, it's the first one we see.
Yeah.
If it's
This here?
that's
Yeah.
it. That's it, babe. Let me check with Beth about.
We gotta figure out where we're gonna put it first.
¿Cómo? Ajá. Okay, okay. When this telling me that we can try to get this and put in the same box as the black coral.
Okay.
Yeah, they were good.
So that'd be box 1B?
The no in the one we just put in the number 8
Quiver 8.
quiver, sorry, in quiver 8, yeah. This have might be a delicate shell, so I will give it a try so we can just pull it off. The shell might be very delicate, so let's see if we can collect them.
So I would go in.
We're gonna try to collect that bivalve, which is first one we've seen, and growing on top of the bivalve are hydroids that we will also preserve.
Once you feel like you have it, a little twist. Just takes as long as it needs to take. No rush. So I would match, you see that angle that that's? Yeah, try to match that with your arm. Somewhere, maybe a little bit more. To the left. Somewhere close to that. Yep, so notice, see a nice straight line with it?
Ajá, ajá. Uh,
No, get it away from that wall. Okay.
still attached.
Just do what you can.
Uh-huh. As you can see, collecting a specimen might take a while. Have to be very careful collecting it and making sure we don't destroy it and that we can get it to one of the baskets or quivers. Como pueden ver, toma su rato cada colecta. Hay que hacerlo con muchísimo cuidado para no despedazar el espécimen. Y después, para no perderlo de camino cuando lo llevamos a alguno de los... Okay. Let's see. We can rescue. No. Okay. Well, we tried. No problem. We tried. We'll look for another one. And but now you got the film of fire there. That's the problem, because you need to be strong enough to pull it off, but not that much to break it. So it's those are hard. Those are hard to collect. Yeah, no problem. Thank you. Well, that happens, especially with some of the organisms are very delicate. So it's a balance between exerting enough pressure to be able to pull it off the rock, but not that much that it will break it. And as you can see, it's very thin line between one and the other. Como pudieron ver, cuesta mucho recolectar algunos de estos organismos. Algunos son fáciles, pero otros sí. Difícil, y especialmente estos bivalvos, que son muy delicados. Entonces hay que agarrarlos con suficiente presión para poder despegarlo de la piedra, pero no tanta como para romperlo. Y esa línea entre uno y otro es muy, muy fina, muy, muy difícil de recolectarlos. Good. Bueno, llevamos el coral negro y vamos a continuar. Continue up to waypoint 6. Continuamos nuestro recorrido hacia el siguiente punto que tenemos, objetivo en este día, que va a ser el punto número 6. Qué más espectacular este lugar. Vamos a continuar la travesía. Vamos hacia otro de nuestros puntos. Se escoge una serie de puntos para cada inmersión, basado en batimetrías que tenemos y con la idea de ir viendo diferentes ambientes. Y aquí nos estamos desplazando hacia la cima de este monte submarino. y vemos sí, una riqueza de organismos, anémonas muy grandes. Hay un crinoideo, octocorales, esponjas, corales negros, poliquetos. Y durante el día se han visto también unas rayas. Una de esas rayas medía 1,5 metros. So we continue going up the seamount and looking at all these different organisms, sponges, octocorals, black corals, polychaetes, little crab over there, shrimp, stars hanging on the octocorals. There we have a sea cucumber. I think we collected one previously. And above the sea cucumber, there's sea pens, more black corals, octocorals. And what we're seeing is the big organisms. As you can see, there are many. We're coming up to some beautiful. This octocoral with a crinoid at the very tip. And this octocoral must be a meter or 2 long. Wow. Puede tener un octocoral de esos. Podría ser si 2, 3. Ajá. 300 años. I'm talking here with my colleague, Alice Cabridi, that studied the octocorals. And we calculate that some of these octocorals could be 2, 3 meters long. And if we assume growth rate of 1 centimeter, we're talking about 200 to 300-year-old animals living on that rock. It started out as a little polyp on the rock and just grew years and years to these immense sizes. Está aquí conmigo la doctora Alice Cabridi, especialista en octocorales, y hablábamos de que algunos de estos octocorales pueden medir 2, 3 metros, y si asumimos una tasa de crecimiento de 1 centímetro al año, estamos hablando de animales que tienen entre 200 y 300 años de estar pegados a esta piedra. Ahí empezaron como un pólipo pequeñito, una larva que se asentó, y ahí empezó a crecer hace unos 200 o 300 años, si no más todavía. Sí, es lo impresionante. ¿Sí? ¿Cómo? Let's check out the one that was on this side. ¿Cuál? Este. El que estoy apuntando. That one over there. Recogieron más plumas marinas de esas. Recogieron más de esas. Ayer. De las plumas. Que se había despedazado. Esas recogieron más.
Ah, de la que salió hace un rato.
No, de esa.
Ah, sí, sí, la tenemos.
Pero No sé, eso ya la segunda colecta.
No, pero antiguo de una vez se marcó.
Ah, bueno, la primera que me enseñaste era
Sí, se hizo pedazos.
total.
Ahora, si sale otro por ahí negro, lo podemos hacer ahora en uno de hojas cookie.
Ajá.
Eso no es lo que tiene que ver.
Mhm.
Ese sí ya lo tenemos.
So we collected this before. That's a sea pen, and wow, qué buena toma. Beautiful. Wow, ¿esos son huevos o qué es eso?
Ay, qué chido. No, esos son penaturas.
Estos penatuláceos, it's a sea pen.
¿Será eso tan chido? Deben ser huevos.
Yeah, we're wondering what those round things are, if they're eggs or we haven't seen that before. Mm-hmm. Y el que queremos ver es un poco más the one we went to see, the other one is a big white one.
No, era más arriba.
Farther up.
That one.
That one? Okay. Zoom into that one. Ah, es un coral negro.
Se puede agarrar y poner con el otro coral negro.
Let's see what sizes it is. Es que me parece que va a ser muy grande. Sí, sí, está. Que está, mide como
30, sí, se puede agarrar.
como así.
Like 10, like 30 centimeters high, maybe. No.
Yeah.
Yeah.
It fits in the clear. No,
It's so wide. So that's
no,
Yeah.
because it folds. It's just kind of flexible, so you can put it in it. We'll do this, but
it's true. The only problem is when we grab the whole thing, we end up holding it by that tip. So there's no way to get it in there to fold that way. So we always have to we have to put it in upside
Yes,
down.
sir.
Sometimes if we can get it stuck enough to get a regrab, then yes, we can flip over and go that way. But a lot of times when we do that with the current, it blows out. So it's not our favorite way to do it. It's just difficult to take something that large and land and put it in the river. We can try. We can definitely try.
no, I don't know if a branch would work for the capital. Como una rama, ¿no? Una rama como a la mitad del bicho.
Like from here?
Break it like there.
Yeah, let's take like here.
Oh yeah.
That would be, yeah, that would be easy
Yeah,
to put it
that
in.
will work out. Maybe here to pick the lateral too.
Sure, we can try that. Yeah.
Okay, thank you.
Excellent. Yeah, this is another black coral.
We are out of coral quivers though, so
Well,
which one do you
wait,
want to put
in number 8, with the other,
Or
with
the
the other,
other
The other one?
with the other black coral.
Okay.
Okay, let's before we collect it, do some images with the laser, without the laser, and then details of the base where it's attached.
There's it.
Okay, there it is with the lasers. Ahí está. I don't know, we'll be able, yeah, this other one is covering the whole fence.
Let me reposition.
Okay. Vemos una estrella de mar, más corales negros. Vamos a reposicionarnos para tratar de colectar una parte de este coral negro. So we're gonna reposition to get a better view of this black coral. Do some video. No, estamos cerca del 6. Ahorita camino entre 5 y 6. Aquí estamos subiendo la cima. Sí. Ajá, exacto. Sí, vamos a... Sí, para tomarlo, sí. Bueno, si hay suficiente sedimento, ¿verdad? Lo que sospechamos es que va a haber capas. Con suerte ni siquiera el push core. Coral negro, dos especies diferentes. Ajá. Con mucho gusto, aquí estamos narrando en español y okay, and they want an image of this one also, just a fixed, very high quality image of that one. Y esta es otra especie de coral negro, another species. We're doing some video, very good images. Yeah, it's only with these vehicles that we have a chance to see these organisms where they live and their natural color and who is associated with them. Like in this one, we see some crustacean attached to one of the branches. Okay. Very nice, thank you. Ahí estamos filmando otra especie. Vamos a alistarnos para hacer la colecta del que nos interesa en este momento. Aquí estamos a 2025 metros de profundidad y con una temperatura de 2.21 grados centígrados. And right now we are at 2,025 meters depth with temperature outside of 2.21 degrees Celsius. And we're getting ready to collect this other black coral and take it, as I mentioned before, this will be taken to the Zoology Museum at the University of Costa Rica, where it will be studied in detail with light microscope, electron microscopy, DNA. Estamos ahora listándonos para recolectar este otro coral negro y como mencionado, todos estos especímenes se depositan en el Museo de Zoología de la Universidad de Costa Rica y ahí van a ser estudiados con microscopios ópticos y microscopía electrónica además de análisis genéticos.
Sorry? Yeah, you're a little out. You can kind of go more direct to it. I think it'll be easier to grab it. It's going to be quite a reach with that angle. So straight on. Yeah, I would yaw right. Come a little bit straighter onto it. Doesn't have to be straight on, but straighter, and then you want it to reach as far. Got it. Yep. Like that. See how that feels. Looks like you're almost there. Yeah. We're going to get a little bit more.
Just below the branch, where it branches. ¿Por dónde quieren el corte? No, digo que el corte aquí o aquí. Yeah, like here. Uh-huh. Yeah, like here.
You're way too low. You're against the rock.
Sí.
Try to use all of them.
Sí, esto va a depender mucho de la identificación con los videos. Como son tan grandes, no se puede agarrar mucho.
Sí.
Yo al menos como de 5 minutos el más cambió 3 géneros diferentes.
Así.
Así que lo que digo es que como hacen para hacer tan pateado, no me da vergüenza hasta decirlo más simple. Escribirles, le estoy preguntando que cómo se identifican los suicidios. Es como una parte de arriba es suficiente. Eso es muy fuerte. No me ha contestado, vamos a ver si me contesta. Para que se sienta incluido.
Sí, está muy bien.
Rotate and try to break it. It doesn't break. Just got to rotate a little bit further.
¿Verdad?
Just gotta keep rotating it, bending it, bending it. Eventually it'll create a weak point where it'll break.
No.
Yeah, this is common with this species where it does that.
Yeah.
Many times we try to sample these, and that's what it does. It's very difficult to get it to cut.
Yeah, and the problem is that
They are flexible.
it removed the
It skins it, yeah.
skins it, that was the word. ¿Qué hacemos?
I mean, yeah, you got a hold of it. Keep trying. There you go. You got it.
Okay. We got a piece. Y un montón de pólipos sueltos.
Sí, ¿para qué quieren? Que también es más de razón que acá, como no pudimos acá una rama grande, se podría recolectar otra. Lateral, tal vez.
Vamos a quedar en la misma, ¿verdad?
Sí, va a volver a romper solo un pedacito. Yo creo que recolectar un pedazo de los patos, que ahora está complementar de esa pedazo, porque se le saca de...
¿Cómo?
No, es que ella dice que agarre el umbretates, pero el otro, por el libro que hay ahí, el que está delante. Pero no sé, mira, es que ese es un libro.
Okay, todos son ahí. Sí, ¿verdad? No.
Catch it in the air.
Next time.
Okay, good.
So we try with another branch. So we move on.
No se podía con el slurp y alargar toda la...
Todo, no es que trataron y se quedó pensado en la manguera.
La vez pasada se perdió, pero tal vez que se aseguren que él...
You're doing fine. I'm just,
No, es que por el tamaño. Sí, se va a quedar ahí, no pueden. No, ya una vez aquí sí,
No pudieron sacar, la vez pasada se perdió.
this one
El
is
que no, el slurp se perdió,
one to get. That's how
que se revenció.
y de algún lado.
Y es lo que yo les decía de ellos.
Ajá, es raro.
No, it wasn't.
Que vamos por otro pedacito, a ver.
Esta rama de este lado que está más
Yeah.
grande.
Yeah, like.
Maybe down there that the branches are more breakable, the ones that are in the bottom.
This one. From there. See if that works.
Try not to pull once you got a hold of it. Just try to rotate on that little pinch point and try and weaken it.
but I'm gonna try for another branch. And as I mentioned before, collecting in the deep sea is not easy, even with these great controllers here, good equipment, and things like that happen. When it's almost in the quiver, there's a little movement and it just flies off. So we're gonna give it another try. Como pudieron ver, no es fácil recolectar que en aguas profundas, es en esta forma indirecta, con un brazo mecánico. Cuando ya lo teníamos, a la hora de tratar de ponerlo en el recipiente, pasó una pequeña corriente y se fue. Para que vea la estrellita. Sí.
Try to do like 90 degree up, 90 degree down. It's going to take quite a few of those. You heard that. All you got to do is now pull very slowly. You see it slipping, then stop. Yeah, so it doesn't look like it's in the blade. It's definitely in the pictures without the blade. You might have to bump up and try to get it in the blade.
Ajá.
These ones are always fun. Yeah. I want to just peel this. Just keep going. It does, it does.
Uh-huh. Yeah, yeah. Yeah. Has to be.
That was difficult. That's one of the more difficult ones.
Okay, we got a good sample. Okay, let's see. We got a good sample. Let's get it in the quiver.
So once you get away from the rock,
Bueno, ya llegó el final de mi periodo aquí en Cuarto Control. Jorge Cortés de la Universidad de Costa Rica. Me despido. Les agradezco que nos acompañen y que estén enviando preguntas y comentarios del trabajo que hacemos. Y aquí seguimos. Estaremos aquí como hasta las 3 de la tarde, hora de Costa Rica. Well, thank you very much. My time is up. We changed the shift time, so now it's 1 hour to give a chance to more people to be here in the control room and speaking to you, explaining what we're doing. And I hope you enjoyed this last hour, and we'll continue for a couple more hours here. So keep tuned. Bye-bye. Bueno, para la próxima hora nos va a acompañar la doctora María Isabel Sandoval, profesora de Geología de la Universidad de Costa Rica. Entonces vamos a oír más historias geológicas en la próxima hora. In the next hour, Dr. María Isabel Sandoval, Geology professor from the University of Costa Rica, will be here at the control room, and you'll probably hear more geological stories. And that's the beauty of this expedition with so many specialists in different fields. You get to hear about a lot, and we get to learn a lot from all of our colleagues. Thank you.
Muy buenas tardes, les habla María Isabel Sandoval y vamos a continuar con el trayecto por este Seamount llamado Tengosed y estaremos colectando algunas muestras, pero principalmente estaremos recorriendo por la primera vez a detalle este seamount o este monte submarino, como se le dice el término en español. Hello everyone, I'm Maria Sandoval. So we will continue collecting some samples, but we are also trying to do and to recognize all the features that we have in this seamount called Tengosed. So if you have any questions about it, about the geology, just let me know and I will be very happy to answer it if I know it, if I know the answer. Si tienen alguna pregunta geológica de estos sitios, me podrían consultar. Si conozco la respuesta, estoy contenta de ayudarles. Aquí conmigo en el control room está la doctora Mayla Guillón. Ella es brasileña, pero vive en Alemania y trabaja para el Research Institute for Sustainability in Potsdam. So here with me in the control room is Mayla Guillaume. She's a Brazilian scientist that works for the Research Institute for Sustainability in Potsdam, Germany. So she will be helping me taking notes about the organisms and the geological features too. So for now, we are trying to get in the coral quiver. Yes, para la pregunta en el chat tenemos, dicen que si son basaltos principalmente, sí, efectivamente son basaltos, vienen desde digamos, desde la Mid Ocean Ridge, como olvidé el nombre en español, disculpen, y principalmente vienen de ahí donde se genera la corteza oceánica del East Pacific Rise, y posteriormente se generan ahí como pequeños volcancitos de basaltos y se produce la corteza oceánica y son principalmente de ese tipo de composición de los mid-ocean ridges. La dorsal oceánica. La dorsal oceánica. Ese es el término en español que utilizamos. No, for the question that they say, what type of formation does have this site? Are there thermal vents? Not for now. That we have, that they have measured in that place is a place that the cold water is getting into the crust. So we can call a recharge spot instead of a outflow or a discharge spot like we saw in Dorado, for example. Tenemos una pregunta bastante interesante que nos preguntan si acá es un vent termal o que si está caliente, saliendo agua caliente, y justamente les comento que en este sitio del Seamount, del Tengosed Seamount, y por eso la palabra, o el nombre de Tengosed es más bien lo que está pasando acá, o lo que se cree es que está recargando de agua fría. Hay una recarga de agua hacia la corteza terrestre y más bien sale caliente en algunos sectores, por ejemplo, como el Dorado. Well, we are almost finished with the sample in the coral quiver. This is an octocoral that we collected, a black coral, sorry. So we will continue then to the waypoint, to the next point that we have to check it out. Bueno, casi estamos terminando con la recolecta de la muestra del coral negro y continuaremos bien, excelente, y continuaremos hacia el siguiente punto para observar observar y ir recorriendo el monte submarino. So we can continue for the next. We are moving to the next waypoint. We have to, and maybe in the way to get there, we can collect some more rocks. Okay, so for the location that we are 200, no, we'll say 200, 150 kilometers away from the Nicoya Peninsula in Costa Rica, away from the coast. So maybe you can just add like a Dorado Outcrop searching in Google. Like that you can, or Tengosed Seamount. And maybe you can find a location of this mission. This is a question, this is an answer for James, that he is asking about the location. Bueno, les recuerdo que acá estamos ubicados a unos 150 o casi 200 kilómetros desde la costa de Guanacaste, de la península de Nicoya, mar adentro. Y si gustan ahí buscar en Google o en Internet, podrían buscar la localidad como Dorado Outcrop o, por ejemplo, Tengosed Seamount o Monte Submarino.
That is a crazy looking map. All right, what do we do? Along this ridge here? sat down earlier, I tried to land out, and I was like, couldn't figure it out. I was running into other people. All these were completely random scientific.
Lo que estamos viendo principalmente es, bueno, este escarpe rocoso, ¿verdad? De rocas principalmente basaltos, con algunas esponjas octocorales. En este caso tenemos un crinoideo. Y estas esponjas pedunculadas que viven, digamos, sostenidas. ¿La crecilla?
Right. Yes. We only have about 2 hours left, and we have a long way to go. So let's just move.
Okay. Yeah. So we have to. Okay. So we have to. So we will continue a little bit faster because we are in delayed. So we will just go ahead for the next waypoint. So vamos a ir un poquito más rápido porque estamos un poquito retrasados con el tiempo y tenemos que llegar al siguiente punto de observación. So we are moving to the top of the seamount. Vamos hacia el tope del seamount en el siguiente punto o acercándonos al tope. What we can see here is there is an escarpment, lava escarpment, and all the escarpment is full of octocorals, black corals, also this stalked sponge, glass sponges. Por ahí vemos una anémona, algunos ofiuroideos, many ophiuroideans. Algunos equinodermos regulares a nuestra derecha. Aquí estamos viendo algunas esponjas de vidrio que les llamamos. So we have here some glass sponges and also octocorals, ophiurids, black corals, and a sea star attached to the rock. This is a black coral, a beautiful black coral. A crinoid there attached to the black coral. Un crinoideo. atado al coral negro. A beautiful sponge there, just with a crinoid too. Una hermosa esponja acabamos de ver con un crinoideo encima. Les recuerdo que el objetivo de nuestra investigación es recorrer por primera vez estos montes submarinos en el norte de Costa Rica y pues conocer más a detalle algunos, bueno, desde la parte geológica, desde la parte hidrogeológica y desde el punto de vista biológico, efectivamente. Y uno de los grandes objetivos de esta expedición era asociar o entender, por ejemplo, que en el monte submarino El Dorado tenemos una gran cantidad de pulpos anidando e incubando sus huevos para en una salida o asociados a una salida de aguas un poquito más calientes de lo normal. Entonces esas serían las zonas que le llamamos zonas de descarga, donde el agua que ha pasado a través de la corteza sale ahí con un poquito más de calor, tipo 12 grados, entre 9 a 12 grados. Y estas zonas, por ejemplo, el Tengosed Seamount que estamos viendo actualmente, es un seamount que más bien le ingresaría a lo que es el agua fría, entonces funciona como un sitio de recarga de agua fría para poder entonces transportarlo a través de la corteza y calentar un poquito esta agua, y esta agua saldría más caliente en algunos sitios. Y por lo tanto, digamos, hay sitios donde vamos a tener mucho más anidación y organismos como los pulpos. Y acá en este caso estamos teniendo pues otra fauna, ¿verdad? Comparado mucho más, octocorales, esponjas, black corals. Y no hemos, por el momento en este sector, no hemos visto pulpos, por ejemplo.
Yeah, if you can just keep the waypoint and coordinates so we don't stop.
A big sponge there. With a crustacean and some ophiurids at the top. This is huge. Un saludo a mi mamá, que está ahí felicitándonos por esta expedición. Hola a todos, a todas, familia, amigos. una hermosa esponja de vidrio con algunos ofiuroideos y un crustáceo encima. También vemos ahí un crinoideo en la parte basal. Les recuerdo que estos dos organismos son familia de la familia Equinodermata o del grupo de equinodermos, tanto los ofiuroideos como los crinoideos. Desde los objetivos de investigación y de los objetivos que tenemos cada uno de los investigadores de esta expedición, pues va desde el punto biológico, por lo tanto colectamos muestras. Las muestras que no son colectadas podemos entonces analizarlas con fotografías, por ejemplo. We'll take a look of this. What is this? Sea-pans. Sí, can we close up to the sea-pans, please? These ones to this. Yeah, please to this. Yeah, thank you. We are getting closer to this to check if it's a sea-pan. No? Could be a no. Okay. We have the expert here. So, Odaliska. So she can help me with this identification. My background is in geology, so I'm kind of lost with this. Looks like an octocoral, right? Yes, it's an octocoral. Yeah. One of the big reasons that we are collecting more corals and octocorals and black corals is because we have the specialists on board that they are studying them. Also the sponges that we have collected, and they sometimes they get like broken in the collection when we collect it. So one of the reasons is that too. Sea-pen, yeah. Okay. Vamos a intentar. Ya, pero okay. So we're trying here to get some push cores. Yes. So does this Excalibur? Or the hit slow? Okay. ¿Y cuál es el Excalibur? We'll try the Excalibur. They say if we have the enough sediment to get some push cores and some misting bottles too. Yeah? Okay.
Let me move a little bit to the left.
Okay, perfect. So lo que estamos viendo acá es un octocoral. How you say in Spanish? Pluma de mar, me dice el especialista Odalis Cabridi. Tenemos aquí algunos organismos y vamos a ver si el sedimento es suficientemente espeso para poder colectar algunos push cores en este sector. Okay.
It looks pretty rocky anyway.
Yeah.
Yeah,
Okay.
you look to the side there.
say that it looks very rough.
It's doable, but
Yeah, it's doable. Yeah. We'll try.
See how deep.
Stay here.
First, let's try with Excalibur.
Yeah, we'll try with Excalibur. Yeah. Okay. So we will try here to look what is the thickness of the sediments, but we can try to collect some push cores and to do some measurements about the heat flow, and also we can take some bottle samples, water bottle samples.
so that wouldn't have been a good depth. So
Either 60 or 30, but 50 if you get some lines. It must be at 60 hertz. So it must be at 60 hertz, and that's why it's it looks pretty rough.
Yeah, it looks very.
Alright, we're way off the seabed of the spot of that right here. So we turn this up. They're trying to do a heat flow probe, but it's just at the angle, it wouldn't get like 3 or 4 of the.
So close to the there, maybe yeah between.
Less rocky, hopefully it'll settle.
Mhm.
All right, so the pilots are trying to find a good spot for this heat flow probe, which I just put up in the upper right there, and we'll show it when and if we decide to deploy that. We just had kind of a little watch shuffle going on here. Got Chris and pilot, Scott, co-pilot, Alex back in the data log in here. Oh.
Okay, entonces lo vemos una de dos y una de cinco. Okay. Entonces si hay push core, tiramos dos. Vamos a dar los cinco. Okay. Okay.
There we go. We're not going anywhere now. Okay.
This is can we close up? No, it's okay. Yeah. Like, yeah. So here we will test if the sediment is enough to get some push core samples and also to get some heat flow measurements. We will see. If not, we will continue with our trip to the top. Mm-hmm.
So here's the infamous Excalibur. Kind of a I don't know. It's used a couple different ways, but right now we've been using it to sort of cement ourselves in the bottom here. And also to test the softness of the bottom.
Con este aparato, el Excalibur, estamos tratando de conocer la profundidad del sedimento y si es suficiente, o sea, esas. Is not enough for the push core? No.
Try push forward,
Yeah.
but that's pretty.
That long, what is it? Yeah, yeah.
I wouldn't risk that. No. We can try push forward and see how it works.
Okay.
Maybe.
That looked like more than 18 centimeters?
Yeah, it looks.
It seems pretty grainy too.
The marks are 10 centimeters?
It's 10. Yeah.
Okay.
Yeah, but
Try that.
Yeah, I think it's 30.
Wasn't sticky.
So we will try to take some push cores here. Trataremos de tomar algunas muestras de sedimentos con los llamados push cores o testigos al vacío, podríamos traducirlo. Okay, yeah. Yeah.
All right, just gonna kind of do a test push core.
Para explicarles un poco lo que hacemos con estos push cores o con estos testigos de sedimento, se toma la muestra y se analiza cada centímetro o si queremos un estudio más detallado, lo hacemos cada centímetro y si queremos pues un estudio más general, lo analizamos todo el sedimento o parte de él y con esto podemos desde el punto de vista geológico conocer la geología, la mineralogía, perdón, por ejemplo el contenido biogénico. En este caso trabajamos con organismos como diatomeas que están en el sedimento, radiolarios, foraminíferos y también podemos conocer la tasa de sedimentación, por ejemplo, si hacemos dataciones carbono 14 o dataciones de microfósiles, en el caso si son sedimentos más viejos. Y también podemos conocer, por ejemplo, las propiedades físicas del sedimento, conocer si es más arenoso, si es más limoso, si tiene más arcillas o no. Y esto es lo que principalmente trabajamos con los push cores. To let you know what we are doing with the push core sediments, normally what we take is the sample and we study through the core. Oh, okay, it's working, yeah. So then we can collect them and to analyze, it could be each centimeter to see the variation of the mineralogy, the minerals, biogenic content, also could be like physical properties, like we want to know the grain size, for example, and also there are some of my colleagues on board, they are working on the geochemistry of the samples and also some experiments with other types of experiments related to the microbiome and bacteria, fungi, etc. Sí, súper bien. Sí. You know, we have to take a look the number. This is, yeah, there's only two. We will take the one of the 2 liters, and it could be 5 or 6. And the other one will be the extra ones, bottles, because they are 5 liters. So 1 5-liter, 1 2-liter, please. Yeah. Also, we will collect some water samples to analyze the acidification and other. For example, we can also analyze the plankton if it's and others analysis that we are doing here with some of our colleagues here. Vamos a tomar algunas muestras de agua. En el caso de estas muestras de agua, pues se analiza lo que sería un análisis de acidificación oceánica. También estamos tratando de ver si se puede o si sale algo de plancton. Y, por ejemplo, se hacen algunos otros análisis relacionados con el agua también. Entonces, en el lugar que tomamos push cores, tomamos generalmente agua también. So here we will collect 8 samples of the push cores, sediments, and each of the push cores will be for different colleagues and different research objective of this expedition. Aquí tomaremos unos 8 testigos de sedimento. Cada uno de estos testigos tiene diferentes objetivos en la expedición para ser analizados por diferentes colegas. Este análisis toma un poquito de tiempo para tomar los push cores. Entonces, les pido un poco de paciencia, ya que estas muestras para nosotros son muy valiosas. So we will take some time to get these push core samples. So be patient because these samples are very valuable for us.
For anyone that might just be joining us, you are watching live footage from the remotely operated vehicle SuBastian, about just shy of 2,000 meters below the surface of the ocean off Costa Rica at the unexplored seamount Tengosed Seamount. This footage is being broadcast from onboard research vessel Falkor 2, owned and operated by Schmidt Ocean Institute. Right now we are trying to take some push core sediment samples. Scott is currently doing a great job with the manipulator, picking these up and then pressing them down into the sediment. And the idea is that when you pull them back up, there's going to be a nice little core of sediment in there.
Entonces les recuerdo que estamos a 1989 metros en este momento, en el tope de, o casi en el tope de lo que es el Tengosed Seamount. Estamos colectando unos testigos de sedimento para el análisis geológico y también microbiológico. Con estas muestras principalmente lo que trabajamos es aquí en el barco. Bueno, trabajamos en este caso, parte de mi trabajo sería hacer unos frotis del sedimento. Se trabajan como un tipo lámina, se agrega un poquito y luego se determina la mineralogía y conocer un poquito sobre la identificación geológica de estos sedimentos. Posteriormente los llevamos a los laboratorios en la Escuela de Geología y ahí trabajamos con más detalle lo que es la mineralogía. También podemos hacer o hemos realizado algunos análisis de fondo oceánico para también con difractometría, rayos X, además de hacer algunos análisis de propiedades físicas en los laboratorios de la Escuela de Geología de la Universidad de Costa Rica. So with the sediments that we are doing right now, part of my job here at the cruise, so is to prepare some smear slides and to describe the mineralogy. And then with this push course, we go to the University of Costa Rica, to the Central America Geological School, to do some detailed analysis like XRD, mineralogy, and physical properties too. Un saludo a mis colegas, a Stephanie, a Elena, a Viviana, que están aquí escuchándome y trabajan conmigo en este emocionante mundo de sedimentos de fondo oceánico.
As you saw, that one didn't go in
Oh.
quite so far, so now they have to shake the dirt out and try again at a different spot.
Un saludo para mis amigos geólogos también que me están viendo, Meli, Claudia, Enrique, de la Escuela de Geología. So we have Bueno, por ahora los dejo, ya vamos a hacer el cambio de turno. Los dejo con nuestro colega. Que también trabaja con sedimentos, Gustavo Ramírez. De la Universidad de Cal State en Los Ángeles. Así que bienvenido, Gustavo. Muchísimas gracias a todos.
Thank you.
Hello, everyone. I'd like to thank my colleague, Doctora María Isabel Sandoval of University of Costa Rica for taking over that last hour. My name is Gustavo Ramirez. I'm an assistant professor at the California State University in Los Angeles, and I'll be joining you for the next hour, which should bring us up to the conclusion of this fifth dive of our expedition. Just to recap what we've been up to here, all of today, we are exploring a seamount called Tengosed, which the name is actually quite interesting. It derives from the Spanish I am thirsty, Tengosed. And the reason for this is pretty practical. Based on thermal measurements, so temperatures taken of the general vicinity of the seamount, it appears to be cooler than other seamounts in the area, and it's therefore hypothesized that water goes into the crust through the crevices in the seamount, and therefore it was appropriately named I am thirsty, if this is in fact a water recharge site. We've been out in the Pacific Ocean, Costa Rican Pacific Ocean, for well over a week now, exploring on the fantastic, really remarkable Falcor 2 research vessel, which is, of course, operated by the Schmidt Ocean Institute. And we bring along in the ship another fantastic piece of equipment called the remotely operative vehicle SuBastian. This incredible machines basically are eyes and arms and nose. It allows us to record physical properties of the ocean in real time, obviously record live video footage, and to manipulate experiments and deployments from a control room in our floating research vessel, and basically be present and active and interacting with biological, geological, and physical samples at the seafloor, almost 3,000 meters below here. So the majority of this dive has involved exploration of, again, this Tengosed mountain or seafloor or seamount, which I should point out has never been explored to this level of intimacy before. All the data about this seamount, or everything we knew about the seamount, was basically based on temperature recordings of the seafloor. So we are the first humans to remotely lay eyes on these fantastic seafloor environment here. Again, Tengosed. We were also the first to explore two previous sites on this expedition already. We explored smaller seamounts, one called Fuente, just a couple of dives ago, and another seamount that is yet to be named. Currently, we refer to as unexplored seamount. The first two dives of the expedition involved returning to a very interesting place where water doesn't enter the seafloor, but actually, completely contrary to that, it exits the seafloor. Very small seafloor rock structure called Dorado Seamount. So we are not the first humans to have visited Dorado with a remotely operated vehicle. That site was extensively studied in a previous expedition close to a decade ago. And we returned there for a few specific, very specific reasons. Our expedition, again, nearly a decade ago, there identified hundreds, if not thousands, of octopus inhabiting the rocky crevices up high on the seamount where hydrothermal venting was observed. So it was very obvious that the octopuses had preferences for spots that were hydrothermal or slightly warmer fluids in the background seawater were shimmering out of the rock crevices. So second thing that expedition 10 years ago identified is that these octopuses were there brooding their eggs. That is, it was largely a community of females that set around their egg clutches and protected them from insults, attacks by other macrofauna in the area. So one very interesting enigma that emerged from that is that the egg clutches, again, hundreds or thousands perhaps of egg clutches, each guarded by an octopus mother or female, the eggs did not appear to have viable embryos as of the last time that people were here 10 years ago. So that was a very that was a salient question that remained when mankind last explored the seamount. When we were there, when humankind last explored the seamount, so upon our return in this expedition, we were able to confirm two incredible things. So we confirmed that some of those eggs had indeed embryos with eyes that appeared to be staring back at us. So people speculated perhaps that the eggs could have been non-viable based on the observations from 10 years ago. But again, on our very first dive during this expedition, we confirmed that the eggs were indeed viable. And a couple of minutes after seeing embryos inside of dozens and dozens of egg clutches, we were fortunate enough, or we were all incredibly surprised to see baby octopus just swimming by the dozens right in front of our cameras. So we have indeed confirmed here, as of our first dive, that those octopus colonies are indeed quite happy there, and that most, if not all, of the egg clutches that we observed were indeed viable. So those are major things to report from, again, our first dive of our Octopus Odyssey expedition. So yeah.
Once this is done.
Yeah.
Move to the waypoints. No stopping.
Got it.
Thank
10-4. So again, we are extremely fortunate to get the chance to do exploratory dives. Most of the time, we just simply don't have the luxury to explore new seafloor sites. There's a lot more ground to cover here. Again, this mountain has never been explored by human eyes before, so it's very important that we cover as much ground as possible. So we'll be wrapping up our sediment push core samples here pretty soon in order to basically get back on the road and cover more ground here and just see what lies closer to the summit of this Tengosed Seamount. Thus far, wherever there's bare rock on the seafloor, we've been looking at a very high diversity of corals. I guess some sea sponges as well, and we've been collecting a number of specimens for scientific cataloging. It's been a very active dive thus far, but again, that's great for the biologists that are part of our expedition. But we do want to characterize the ecology of the area too, and in order to do that, we do need to cover as much ground as possible and get as much video as possible to know the diversity of potential environments that exist in this, again, previously unexplored seamount. And that is going to be the goal for the remaining hour of this dive. This will be the last samples probably recovered here. And we're gonna hit the gas here and try to have a really good look at the rest of the area.
Should be good right there.
Chris, are these 3D printed, the little collars up top?
Again, my name is Gustavo Ramirez. I'm an assistant professor in microbial ecology and bioinformatics at the California State University in Los Angeles. I am joined by Myla Guillon, Dr. Myla Guillon, who's a research assistant at the Research Institute for Sustainability in Helmholtz, Potsdam, Germany. And I am also joined by the ROV pilots, Chris and Scott, from the Schmidt Ocean Institute, and our media correspondent, Alex. My primary goals for this expedition are to explore the microbe rock interactions that occur at these incredible seamounts. And in order to do that, I've deployed a set of substrate colonization exposure experiments at 2 sites during this expedition thus far. One of those sites was the Dorado Seamount, where we did our first 2 dives. And that's basically the only site that had been previously explored that we will visit during this expedition. And we knew that there was hydrothermal venting there, and that's the site where I just described the octopus brooding behavior just a few minutes ago. So I was lucky enough to leave behind a series of experiments, very simple experiments comprised of sterilized substrates right at the site of hydrothermal venting, in between a couple of brooding octopus females. And my hope is to come back and personally collect those samples 6 months from now. So my rocks, again, initially sterile at time 0, should accrue biomass on their surface. And this biomass should largely be derived from the unique microbial communities that emanate to the seafloor from the hydrothermal fluids. Again, I have a control experiment with the same exact substrates deployed at a site where there is no hydrothermal venting. And by directly comparing the microbial communities that colonize the rocks under the hydrothermal venting regime and comparing those to the microbial community that colonize my originally sterile rocks deployed at a place without hydrothermal venting, I should be able to say something about which microbes emanate in those hydrothermal fluids and how they change the composition of the rock and ultimately how they interact with the environment. So that's like my area of specialty and my niche sub-experiment out of the many different experiments and important scientific questions that are being asked by all the scientists that are part of this expedition.
So I've just put this view up because I love watching the main camera, the science camera. This is sort of a coordinated effort. We've got Chris controlling the camera and then Scott controlling the manipulator. And of course, Scott needs to see where the manipulator is. So Chris has to kind of follow along and zoom in and make sure that Scott's got the best angles that he can. I really like that one.
I'll be right back. Go more, outboard and port. Look right there, you can take it. That's a good angle there too. Yeah. Unfortunately, it's not helpful on many of those. But it's good when it's good. Yeah. Jill was telling me that carrying around fish boards yesterday. Or she didn't. We don't just like set it down. That made it a lot easier. Oh yeah, that you had free reign and easy grabs. Fortunately, you carry it around. So we're going to do Niskins after this. You want to do them on seabed or you want to seabed what? For the Niskins.
Suppose that we do it here.
Well, right here we're doing it while we're still on seabed or you're going to float out a few meters.
I believe that the closest to the seabed would be best.
Alright. Which makes it easy. Makes it easier.
What was that?
So it makes it easier for me.
Sure. I guess they do want the bottom waters closer to the seafloor, the better. I would just be hesitant if there was like a giant plume or something. Seems like this was pretty normal, right, in terms of plume generation and push for collection. Yeah. Okay. What are the viewers seeing online? Just this right now? Or that?
They're seeing this image right here.
Yeah.
Earlier it was had a picture of one of our this camera right here is back up.
So it's this.
Yeah. All right.
So
Cool.
That's the back of the science camera.
Got it.
of course, one's got to break the tape.
I don't think that there's any
Is that the bungee that's in it?
any thing expanding in the sediment, so it should be an issue.
Yeah.
Usually that's essential to have goes on there whenever it'll expand and fly. So you said that I mean, we'll put it away real quick.
For those of you not catching that, you may have seen these caps pop off a couple times, and the general consensus right now seems to be they're not as important for this dive, but there has been times that ROV SuBastian has taken push cores of sediment that has gases in it, frozen gases, and as you're coming up to the surface, those will start to expand and basically can shoot the little core out if you don't have something to hold it down.
Ideally, we'll put it into a bio box so that it won't have a chance to float away. With the bio box that's open.
There's been times they've been looking at methane seeps, and that methane can kind of be frozen into a
We're just going to hit the waypoints.
like
Just
a
gotta move, yeah.
ice matrix,
Is there a
basically.
50 oil box?
Or if there's just any gas in there at all, that'll also expand.
Split into 4. I don't believe it's anything. Let's look at it and see. grab that other width that came off as well. I was a little suspect about that one. I put it on, but
There can be gases, obviously, at the hydrothermal vents, but sometimes there's other areas of gases coming out. Methane is a big one. Actually, I want to say Okeanos, the NOAA exploration vessel, Okeanos Explorer, is up in Alaska right now doing some work up there, and I want to say they were looking at some methane bubbling out of the bottom not too long ago.
it's gonna get real dirty real fast. So.
Was the missing the spoiler?
No, it was not.
Yeah, I mean, looking at this plume, maybe we will ascend a bit or just move.
Move up to it.
Yeah. Yeah, yeah, that's coming out. But I'm going to ask him to just navigate a little bit away from the plume.
Oh, of course. It's also pointed out Ocean Networks Canada has been doing work, and they were looking at some methane seeps. And it's something that ROV SuBastian has also done as well. It's just not something that we have encountered here. So even though those caps, we like to put them on when they pop off, we're not currently too concerned about this sediment. It doesn't seem particularly gaseous.
Go for it again. It's good practice.
Going for the interest of time.
Yeah.
I mean, my only issue with not putting that one on is that when we bring the rail in, it'll get caught.
So we are, for those of you just joining us, we are just wrapping up a set of collections for sediment push core samples. The sediment here really has the potential to tell a number of incredible stories. It can tell us the origin of the sediment, if it was made in the ocean column or if it drifted in from the land that's not so far away from here. As a microbial ecologist, it can tell me a lot about what metabolisms and what microbial communities inhabit these sediments also, and what biogeochemical cycling, how they contribute to nutrient cycling in the environment. It's also something that can be resolved from looking at the microbes that live in the sediment. Again, the goal for the 40 minutes or so remaining in this dive will be to basically do a survey, just a visual survey of as much ground as we can cover here. Okay. Again, these bungee caps on the sediments are a prophylactic measure just in case that we collected sediments that contain materials with gas that could potentially expand and basically shoot out of the holsters of the ROV. Seems unlikely to be the case here, but it's good practice for the pilots to keep their protocols consistent. I'll look at the chat in a second. We want to be extra careful, of course, not to leave behind any materials from our experiments. So we're making sure to pick up the two parts of the sediment core system that broke off when our bungee broke just a second ago, and make sure we take that with us too. Want to leave this wonderful environment as pristine as possible. That's the goal here. So I guess Putting this plastic piece back in our ROV will then give us a green light to move a few meters away from this giant plume and then collect something called a Niskin sample. So these are open cylindrical containers that are that the ROV has on the top left corner. What happens there is that those cylindrical containers have tops and bottoms, stompers that can be triggered here to bring back water from the depth where the stompers close. That's guess that's the best general description that I can give for something called the Niskin bottle. We want a general sample now of deep water, and in order to do that, we will just slightly move a few meters away from this plume that's been raised by our manipulation of the push course at the seafloor, just a few meters away, to collect more representative background seawater to then bring back up to the surface and filter.
All right. Can you put Excalibur away before we can? So we just get it somewhere where you can see. And then with that, we're gonna be super messy for a minute. But it is.
How many discos do you want to fire?
We're gonna do one small one.
One small?
Yeah. She told me one 2.5 and 1.5. Yeah. Okay.
One of each?
Yeah.
Okay. One of each, Scott. One small, one large.
One small, one big?
Yeah. Okay. That's just where we were, and there's similar corals as to where we just were, so this should be very much the same for water culture. Now, hopefully, X-ray won't follow us. Before I do that, though, before chat a little bit. Sorry. All right, just so you have a better view. I'm gonna come down.
Again, for those of you that are just joining us, my name is Professor Gustavo Ramirez from the California State University in Los Angeles. We are exploring a seamount that's never been seen by human eyes before. We've been at this site all day today. We've explored two similar sites that were completely unexplored previously in this expedition, and visited a third site that was explored before that hosts nursery of octopuses. Seamount ecosystems are quite fascinating. Again, a lot of them are underexplored. I guess an interesting fact here is that even though the seamounts are not particularly large, they look like small pimples on a bathymetric map of the seafloor. When you add up the surface area of these seamounts spread all over the world, if you add up their surface area, they specifically for the ones that protrude more than 1 kilometer tall above the seabed, their surface area is a total of 30 million square kilometers, which is a pretty big number. It's a very impressive number. I Googled what that was just a couple of days ago, and it turns out that that is the surface area of the continent of Africa. And again, we are beginning to explore just more or less an environment that represents a surface area of this planet equivalent to the continent of Africa. Just so happens that this environment is at the seafloor. And before our Octopus Odyssey expedition, all that was known about this, again, worldwide collective environment equivalent to the surface area of Africa, all that was known to science in this level of intimacy was more or less in a surface area that was only equivalent to a football stadium. So that is something that blew my mind, more or less.
Good. All right, we'll do the same thing on that.
Good to go? Yeah. Okay, so I guess we've got about half an hour here. Yeah, and we don't want to stop. Yeah, 2 more waypoints, right? Yeah.
Hey, on the bridge, we can continue on that move to the waypoint.
Now we're going non-stop.
Yeah.
Space trucking. We're on the way.
I don't get distracted and look at anything.
So it's 1 and 6, is that correct?
1 and 6.
1 and 6.
1 and 6. Or 1 and 5. Sorry. 1 and 6.
5 was in this fire, right?
Okay. 1 and 6 and 5 was in this fire.
5 was also fire?
No.
Missed. It did not trigger.
It didn't trigger. Got it.
Okay.
This is just cool. How fun it is.
All right, folks. I'm going to put up the map real quick here. Just so you can sort of see where we're headed. So we are leaving waypoint 6 down here. And we're trying to get as far as we can up towards waypoint 9. So we are just booking it now.
We are going full speed ahead now to make sure that we have as much of a look as we can with our remaining 30 minutes or so of this dive of this ecosystem. As we swim across this Tengosed Seamount, I guess I'll give the audience a background story here of like why we think these seamounts are so fascinating. So basically, a seamount represents part of the crust, or the first layer of the lithosphere, that protrudes above the sediments of the ocean. Sediments are pretty impermeable, so you can almost think of them as like plastic sheets that separate water that's in the regular ocean that we know, right? Which at this place here in the Costa Rican Pacific is about 3,000 meters deep. Again, that's a regular ocean that we can see from the surface. That entire ocean is separated from a ton of flowing water beneath it. And that is, again, separated by sediment. Wherever the first layer of the crust protrudes or comes out of the sediments, this crust is porous, and water will either enter or will exit. The points where the water enters the crust are called recharge zones by hydrogeologists. The points where the water exits the crust are called hydrothermal exit points. The physics behind this is, to my limited understanding, somewhat simple. It's just simply temperature differences of the rocks that drives this what they call hydrothermal siphon. this hydrothermal siphon, right, a slightly cooler side of a seamount will become a recharge zone, meaning water will be denser there and enter the crust. Will enter the crust and flow underneath a layer of sediments. And these sediments can be hundreds of meter thick, kilometers in some parts of the ocean. But again, on average, I would say 100 to 300 meters thick. And underneath them, there's this network of porous basaltic rocks that again represent the first layer of the lithosphere. If those rocks protrude, they become seamounts like the one we're exploring now. And if the seamount is cold, they, because of physics, allow water to enter the crust. So I guess to keep this long story short, the entire volume of the world's oceans, so the ocean that we're familiar with, it comes all the way up to the surface and touches the atmosphere, that entire volume cycles itself through this subsea floor network of basaltic aquifers, entering where the rock is cold and then evolving through its rocky path under the sediments, evolving. It evolves thermically, generally tends to get warmer, or I should say it always gets warmer. It evolves geochemically, the water interacting with those reduced rocks under the sediment change its chemical composition. And most importantly for me as a microbial ecologist, it evolves biologically. So the microbial communities that live in bottom water that enters the crust are completely changed to different dominant species and relative abundances of those dominant species in those same fluids when, again, after getting warmer and changing in chemistry, when they emanate at warm sites of a hydrothermal discharge. So that's really the interesting, or the most interesting, and I guess broadest possible view of why exploring seamount ecosystems is extremely important, because ultimately the seamounts are the conduits for the entire ocean to evolve thermically, geochemically, and biologically. So again, this one cycle of all the world's oceans through this environment occurs approximately once every 300,000 years. Again, that doesn't seem like, that seems like an incredibly long amount of time, but given the fact that perhaps Homo sapiens has been around for couple 2 million years, this has happened a number of times in the same length of time that our species has existed. So it's pretty fast in geological time. Yes, sir.
That's fine. So I think Adam said we can stay on bottom until like between 3:15 and 3:30.
Oh, fantastic.
Because we're getting shallower and shallower.
Yeah.
So
Hey, there's a rock.
So if
All
you
right.
want to stay on past 3,
Yeah, that's fine.
if you don't, just text me and I'll come in.
Yeah. Okay. Perfect. Yeah.
Okay.
All right. Thanks. So as we've been climbing up this seamount, we are, of course, in a shallower depths than we were when we started our survey at the bottom of the seamount. So that means that our ROV recovery time, how long it takes for the submarine to leave the seafloor and come back up to the ship, is significantly shorter. So we've just been informed by our chief scientist, Dr. Beth Orcutt from Bigelow Laboratories. We have been informed that we are clear to continue to explore an additional 25 minutes to half an hour. So, yay.
That doesn't mean I get to stop about this stuff.
No.
Big crap. We're still not stopping, unless
Yeah.
I assume
No,
we'll break
still
for
not
octopus.
What was that?
We'll break for octopus.
Okay, yeah, we'll break for octopus. That's right. We'll break for octopus. But apparently for nothing else.
Another skater or shark. Don't let them stop to look at it. Stop.
Some charismatic macrofauna here. Deep sea fish. We've seen a couple of those in previous dives in the area.
Oh, don't celebrate. I'm not making the rule that we're going to break for octopus. That's just my assumption. The chat's like, oh yeah, octopus.
I think we could definitely break for venting. Yeah.
Mm-hmm.
Yeah, that's
Yeah.
But again, that seems extremely unlikely at the seamount. Yeah. This is the cold one. Tengosed.
This is the cold
Yeah.
seamount, yeah.
Yeah.
That's the thirsty one.
Yep.
Yeah.
On the bridge, could you speed up to 0.3? Yeah. Some larger fish. So we are passing waypoint 7, or we're officially past waypoint 7 now, on our way to 8.
How many waypoints do we have until we reach our summit?
9's the end.
Number 9.
Let's see what we can do.
Number 9. It's twice.
Answering Kat D's question in the chat, I definitely know Dr. Eric Wood. He's director of the new Urban Ecology Center at Cal State LA. Happy to send them your regards, Kat. Looks like we're seeing an explosion of life at the seafloor here. Again, I am a microbial ecologist more than a marine biologist, but I mean, clearly some sponges, octocorals, shrimp, very cool looking fish. It really seems that as soon as the geology changes, the biology changes, and that's the general theme, I guess, first observed by explorers a long time ago. Potentially that basic observation that we take for granted today, such as tree lines on mountains, you know, that are analogous to what we're seeing here, can be traced back all the way to Alexander van Humboldt's expedition around the world. So yeah, we are confirming his suspicion that geological landscapes tend to co-evolve with biology. We've picked a number of interesting geological samples that will be used, I guess, directly applied for geological analysis, but also for microbiological analysis of the composition of the microbiome that lives on the rock on this dive already. So again, our goal for the next 40 minutes or so is to simply do a video survey of this previously unexplored seamount called Tengosed. So we're just very quickly flying through the area and capturing high definition video that geologists back on shore will surely explore for many, many years to come. Again, we've done a number of animal collections as well, octocorals, black corals, and sponges during this dive. All of these collections are done under strict permitting rules with the Costa Rican government, or the stewards of this majestic seafloor habitats. You might see two small green dots on the screen there, and those are two lasers that allow us to measure things at the seafloor, to have a scale of things at the seafloor. They are 10 centimeters apart.
I'm gonna split out to get that best chance to catch up.
Yeah.
So we're pretty far away.
Yeah.
So
Yeah.
this will get some of this stuff, so
Got it. Yeah. Our ROV has been hitting the gas here, covering as much ground as possible, and we are tethered to the ship, the research vessel Falcor, about 2,000 meters, right? 1,935 meters above the ROV. and we've only got so much line, so we do have to wait for our ship to come closer to us so that we can get a little more slack and then carry on our way. So that's what's happening here. In the meantime, we are looking at some of this incredible rock formations here. Again, we're seeing octocorals there with some symbiotic fauna. And maybe see a crab in the background. So again, there's just an explosion of life as soon as bare rock is available on the seafloor.
Yeah, it's pretty.
So as mentioned, our depth is at 1,941 meters, but the amount of umbilical that's actually paid out from Falcor 2 currently just over 2,000 meters. So there's like 60 meters of, difference there. So it's something where we don't want to have to pay too much out. So it's better if Falcor 2 could just keep up. Yeah. Okay. Yeah, so you can do that. Chris just said we're about 120 meters away from directly underneath the vessel.
We just need to be here. We need to make sure that
So there's certainly an angle there. But that's also one of the things you keep track of is like how much is paid out versus your depth. Yeah.
some more seafloor critters here. Is that a crab or a lobster? Crab, lobster, what do we have? Looks like a crab, yeah. Flying shrimp. Yeah. Yeah. Crustaceans, yeah.
Sitting there just waiting. Bring it. See that in a second.
Nice.
What is that? Is that a rat cat? No, fish.
So while the macrobiologists are amazed and awed by the corals and the beauty of the sponges and fish, in my mind, I am just thinking about the different microbial species that could be living on these rocks. Previous studies of microbial or microbiomes associated with seamount rocks have been done at the Dorado Seamount, the site of our first 2 dives during this expedition. So shout out to my colleague, Mike Lee, and who led that study, along with our current chief scientist or co-chief scientist, Dr. Beth Orcutt. The really, really interesting stuff's happening on the surfaces of those rocks at Dorado. So. Oh, wow. Is that a mouse? Or is that a vitamin?
Yeah, a little urchin.
Bigger.
Well, that's true. Yeah.
Do we have the
It
lasers
is
on?
quite
Or is
large.
it-
Yeah, the laser is above it.
Okay.
Yeah, lasers are just
Oh,
above
I see
it.
it. I see it. Okay. Sorry, they blend in with the green there. So we have a sea urchin here. and the lasers are right on it there, so it's slightly bigger than 10 centimeters, perhaps 15 centimeters.
Could be.
No worries, buddy.
Another
Beautiful.
Whoa.
Wow. Is this a skate? Yeah, right.
Yeah, there's another skate.
Yeah. Deep sea Pacific skate here. This is perhaps the second or third one that we've seen on this dive, right?
Second one that
Second one, okay. And I believe the first one that we saw was close to 2 meters in size. Yeah, so.
That first one was female, I believe. This one should be male. It has the claspers, which are those longer structures underneath the tail that you can see there. And I don't think those were present on the one we saw earlier, despite its large size.
Not running anything, I feel bad.
Aw.
Try.
Yeah. Cool animals.
We'll leave you to your
Huh. Wow. We'll get a fish expert here to explain to us, I guess, mechanism for locomotion of these skates, right? Seems like they're flying blind on the seafloor. They must be able to sniff their surroundings, though. So
Yeah, their eyes are up top, their mouth's on the bottom.
Okay.
So basically launch, and then time that comes in from launch.
Yeah.
And then
just seems to me that they must have a mechanism for chemotactic detection, right? Smelling their environment more than seeing it.
Hmm.
And I don't know if they're a bit spooked by us coming. They might just be bumping into things, moving faster than they normally would, smelling the currents or something. So they're very interesting behavior.
So notice that a lot of the things that I followed, sharks as well.
Mhm.
We follow a shark around for a while.
They just bump into
They
stuff.
rocks.
Yeah. Yeah. So I think that they see through smell.
Yeah.
And if they move too fast, they may not be able to interpret what they're smelling correctly. Yeah.
They just sense something weird.
Yeah. Oh, yeah.
It's like,
Oh. Had a little shrimp there fly right in front of our camera. We are getting pretty close now to our waypoint number 8. And in the couple remaining minutes of this dive, it seems that we might be able to make it to waypoint number 9.
luckily, Adam will be pushing me a little bit. And he's the person you just have to convince to sit down. So
To what?
he's the one you have to convince to stay down.
Yeah.
Suspect, come on, we're right there. Sit right. Don't tell us something. I got it.
A lot of those polychaete shells around, it looks like. We saw these wider polychaete tubes further down. It looks like there's a lot up here now. A lot. Oh yeah. Whoa. 25 centimeters probably at least. Yeah. 12 inches or something for folks not handy with their metric. Another one. So many worms. If you're just tuning in, we are on the last little leg here. We're on our way to waypoint 9, currently passing waypoint 8 on Tengosed Seamount. Currently 1,910 meters below the surface of the ocean off of Costa Rica. And you're watching live footage from remotely operated vehicle SuBastian. We're broadcasting this from the surface vessel, research vessel Falcor 2. And we're just making our way up this seamount.
Unless it's something that you just have to do.
All right. Looks like Adam just hopped in the pilot seat here. We've still got Scott as co-pilot.
Yet another one of these long-tailed deep-sea fish. We seem to have encountered a pretty healthy population of them on this site.
Yeah, a lot of rat tails, I believe, compared to the eels that we've been seeing on the other outcrops and the like.
Again, we are continuing our Tengosed Seamount survey here. One more time, a seamount never explored by human eyes before. Wow, that's a wonderful shot.
Let me get the lasers off for a sec.
Yeah.
We're just sitting here.
Wow. Can we get a screen grab of that? Oh, okay.
Good looking fish.
Waiting for the ship to catch up? Yeah. Okay.
It's push cores are like sometimes you just flow with it. They're like easy, and sometimes it's just a freaking struggle the whole time. How are these? What is the struggle? It was 50-50. Enjoy. See you in the morning.
Yeah, that's brilliant.
Thanks, Scott.
Hey, cheers.
Yeah. We continue to glide over this surreal alien landscape, and I mean, every second of these dives, throughout the entire expedition, I've just been mesmerized by how alien this environments seem. Yeah. yet how familiar some of the behaviors exhibited by the animals are. Again, with the main example there being the octopus mothers or octopus females brooding their eggs. It's weird. To capture some of these human perspectives on all the science, all the complex technical science that's being done on this expedition, we do have two artists at sea that are joining us. So that's this is my first expeditions with artists at sea, and this is Carlos and Michael that are on the ship with us now, and they've been working away nonstop at generating new art pieces that are inspired by some of these emotions that can emerge by at the forefront of exploration. So this is again another unique thing that that the Schmidt Ocean Institute has allowed us to or wants us to focus on here. So we have two artists on board with us. I'd like to remind the audience that in expeditions a long time ago, artists on board were actually mandatory, right? All of these explorers from the 1800s needed to document their biological observations, and that could only be done through sketches. So there is a rich history of art on board exploratory research vessels, and again, here on the RB Falcor 2, we continue that history by having our two artists artists at sea with us today. Oh, that looks pretty cool. We're coming up on the tail end of our dive here. We're gliding past waypoint 8 towards our final waypoint, scheduled waypoint for this dive. And again, the goal here is simply to capture as much video of the seafloor as possible. We suspect this seamount, Tengosed, as for those of you Spanish speakers may suspect, to be a site where water enters the seafloor. I am thirsty is the Spanish translation for Tengosed. Again, this water entering the seafloor here is a hypothesis that's derived, as far as I'm concerned, from temperature measurements in the area that identify this large seamount as a cold one, meaning that water will be dense here and in fact enter the crust through these rocks. But yeah, are those sea clams or aqua corals?
Yeah, this is black coral.
Black corals, okay. Seeing a colony of black corals here, along with some octocorals in the background too. And wow, something that appears to be a sponge. And of course, a pretty pronounced slope by now. We continue to rise along the seamount.
This might actually be the summit.
No kidding. Okay. ROV pilots here are informing me that we might have reached the summit of the seamount here. There doesn't appear to be a place higher in elevation. This is here, there somewhere.
Yeah, unless it drops down. You know what? Let's drop down and continue on.
Yeah.
There might be a dip in here, but I definitely see this is what are we at? 1895.
Are you on the eastern side or western side?
This would be on the southern.
Southern. Somewhere there.
Have to zoom out here. Yeah, like.
Oh, okay.
Oh, sorry. This would be the eastern side.
Southeastern side. Okay.
Yeah. You can, and then, actually, let's get the tone moving again. I think we will drop down and just see if that other side is higher.
We have yet to confirm this, but there's a good chance that the ROV has, in fact, summited Tengosed Seamount. So again, this seems to be a gigantic structure relative to the other seamounts that we've explored on this expedition. Dorado Seamount, where hydrothermal fluids emanate from the seafloor, is just slightly bigger than perhaps 1 or 2 football fields, as far as I'm concerned. And this place here is just massive. And stand by for confirmation of whether or not we've summited this. It's a drastic change in the geology that we can see here.
You're right, because I think it drops down into this little gully, and then I think it actually is going to climb. What did we say? 1895?
A couple meters to go.
There's another.
Okay.
Another little summit.
Okay, so we have yet to reach the summit. There should be another small summit in the area here. Unbelievable landscape continues. Just hover right over another rat-tailed fish. Another 2 of them. There's one just to the left of the screen there.
One big sponge.
That is gorgeous, yeah.
Yeah. Something long in the background.
Yeah, what is that in the background? Does not seem like a ratfish. Rat tail.
No.
Maybe an eel?
It's much pointier than the eels we've been seeing.
Wow.
What is this?
Yeah. So it's my first time at the helm here, so let's. Oh wow! Zoom in.
Whoa!
Wow. Well, go ahead and zoom out a bit.
Yep,
Yeah,
just kidding.
there it is.
There we go. Just let it drift. Oh. Spot on, guys. Wow.
Any idea? What do you have there? I don't know.
All right.
It seemed really pointy.
Yeah, you're right.
Yeah. I'm not sure what that last fish was. We've got a hypothesis here that it could be a cutthroat eel, but hopefully our high definition camera footage will allow deep sea fish experts to properly identify it.
Too pointy for that.
Too pointy, right?
Yeah.
Yeah.
There's like duckbill eels or something like that.
Someone in the chat spoke about a sorcerer.
Sorcerer eel.
Oh, okay.
That's chat's got some.
Yeah.
Some in there.
Oh, wow. That start, really nice
Oh,
start.
okay. Yeah, sorcerer eel's a type of the duckbill eels.
Oh, okay. It's that long thing in the octopus. Yeah. And what do you think? It's like 2 meters? Yeah, we suspect that 2 meter structure that we just saw there, it's very likely an octocoral. We are on our way to our final waypoint for this dive, waypoint 9. And we came across a pretty high point of elevation on this Tengosed Seamount that we thought could have been the summit, but we may arrive at an even higher spot. So the legitimate summit may still be to come.
So 1915. So what did that before? 1895. So
I think that's correct.
20 meters was. So we ascended a little bit. Now in the future, there's a little bit of a.
Oh, we got a couple more fish. Let's have a look at these.
Little tripod fish, different type from the ones we've seen now.
Wow. Yeah.
So this one has little sensor rays reaching much further forward. You can see them off the head barely. They're reaching out into the water column.
Can we get a laser on that?
Yes.
Okay, so got a smaller species of tripod fish. We've seen tripods, I guess, in 2 of our previous dives, and they were significantly larger. So again, we hypothesize this is a new species, right, rather than a juvenile.
It's a different species
Different
from what we've
species.
seen, but
Okay.
I've seen these before.
Okay.
Yeah. I mean,
Wow.
that's not to say it's not something different. Oh, another of the eels.
Right.
Oh, man.
Gorgeous.
We could get lasers off just for a moment.
Yeah.
Might.
Okay.
It's another type of
Yeah, it's another of the duckbill eels
Duckbill eel, yeah.
of some sort. Yeah. The black sorcerers are all over in the Atlantic, I think, but it's a duckbill eel of some sort. Too cool.
Few minutes that we've got left of this dive, I will take some time to make a Spanish language summary about all the action that we've experienced over the last hour and 10 minutes here. Bueno, me llamo Gustavo Ramírez, soy un profesor asociado en la California State University en Los Ángeles y tengo el placer de estar en el buque de investigación Falkor 2, un buque que es parte del Schmidt Ocean Institute. Nos encontramos en las aguas del Pacífico costarricense estudiando montañas marinas, montañas submarinas. Estos son partes de la litosfera que salen más altas que el sedimento y estas partes de la litosfera tienen propiedades físicas que hacen que el agua corra de una montaña a otra. Si una montaña es un poco más fría, el agua es más densa, y si hay otra montaña donde hay un poco más de calor, el agua es menos densa. O sea que se hace un tipo de circulación llamada embudo térmico que circula todo el agua del océano una vez cada 300,000 años. O sea que es algo interesantísimo. Bueno, estamos explorando una montaña submarina que nunca se había explorado antes. Los ojos humanos no la habían visto. Se llama Tengosed. Y bueno, se llama Tengosed por lo mismo que acabo de describir, que es una montaña relativamente fría que se supone es una área de recargo donde entra agua agua del océano a la litosfera y corre hacia lugares donde la litosfera es un poco más caliente o lugares de menos densidad. Sí, y algo extraordinario, es un paisaje alienígena. Es como si estuviéramos en una de las lunas de Júpiter y bueno, la meta para este buceo con el submarino SuBastian era simplemente explorar esta montaña submarina que nunca se había visto con ojos humanos antes. Saludos a Ignacio. Sí, creemos que hacer esta expedición bilingüe es súper importante, ya que estamos en las aguas territoriales de Costa Rica y este es un recurso increíble para los costarricenses, ya que más del 90% de su país es el lecho marino profundo. Y bueno, más allá de eso, el océano es algo que nos pertenece a todos, es un patrimonio global y es importantísimo que lo estudiemos juntos en operaciones multinacionales para que podamos entenderlo juntos y podamos preservarlo juntos.
That actually, I think it's just a shadow in the picture.
Got it, yeah.
But I think what I'm doing is that's about 10 or 15 minutes. I'm just going to
Yeah.
run to this little, you see this bathymetric kind of circle?
Yeah, yeah, yeah.
We'll just see if that's another bit higher. Like
Sure.
85 was a minus, but we're kind of ascending here. So.
All right. Okay, so I've just been informed by the ROV pilots that we've got a suspected summit point here. We've got a few minutes left on our dive. Made it to waypoint 9. And we will follow the topography of this mountain to see if we can climb higher than where we've been before. And we are flying over just a completely covered seafloor with brittle stars. Again, we hope that this footage that we're capturing will spark deep sea ecology conversations among some academic circles and the general public for years to come. the final minutes of this dive, I'd like to come back to this. Concept of oh, beautiful. It's a couple of floaters there. Why we think exploration of the seamounts is important. Again, this hydrothermal siphon process that I described a few minutes ago cycles the entire volume of the oceans through the subsea floor crust once every 300,000 years. It's an incredibly important process for the net equilibrium of chemical, physical, and biological equilibrium of the ocean. Of course, the chemistry of the ocean ultimately determines the chemistry of the atmosphere and is ultimately, again, responsible for super important things like the oxidation reduction state of the entire planet. When the oxidation reduction state of the planet's changed before, the entire course of evolution, both at the macro and micro level, has also changed. So it's this really large, grandiose concept of the coevolution of the biosphere, that is all the living entities on this planet, and the geosphere, this gigantic rock, wet rock that's more or less our spaceship, you know, that I'm ultimately interested in. So that is really the big idea that drives my interest in the exploration of these extremely remote environments that again play super important roles, and where I believe the microbes actually play one of the most important roles in the evolution of the chemistry, physics, and biology of the ocean, which leads to the chemical stability of the atmosphere and the oxidation reduction state of the planet as a whole. So that's one thing I wanted to say before coming up to our at the end of our dive here. Again, we believe we had reached the summit just a few minutes ago of this pretty big seamount in the Costa Rican Pacific. However, we are just following the topography here in the last couple minutes that we have left to see if we can go higher. Yeah. So this seamount again has never been explored before. We are the first people to lay eyes on the beautiful alien landscapes that exist here, and that is something that's definitely exciting to share with the rest of you live online from whatever it is that you're joining us. That's more than 2 meters. Yeah. Some of these towering octocorals, you know, just so unexpected, tall and majestic. It's I guess they remind me of driving through Arizona and seeing the saguaro forest. So that's basically what I feel I'm doing here. Yes, extremely tall corals. Again, remind me of giant cactuses or saguaros that make up cactus forests in the middle of the Sonoran Desert. And again, the height of the seamount, topography, geology, they're all, as the background changes, as the rock changes, the life around them changes too. So it's this very intimate connection between geology and biology. That's the ultimate goal of what we're trying to do here with a multidisciplinary group of scientists that make up the science party for this Octopus Odyssey Expedition. Lisa says cacti are rad. I believe that they are, indeed, yes.
1897, so 2 more meters and we'll be even higher than the last.
Okay, all right.
False sign.
Skate egg.
Yeah. Skate egg.
Right down here. Been seeing the skates. I was wondering if we'd see an egg right there. Got ourselves a skate egg. We've been seeing the skates around. We've seen a male. We've seen a female.
There's the
This
egg.
egg, yeah, it looks like.
Looks like it.
Yeah, it looks like it's in pretty old shape though. It might be.
Can we get the lasers on for a second?
Might be what?
Might be hatched out or something. They stay in there quite a long time now.
You know what it is? It's a skate egg. I didn't know that.
Nope, there's another one.
Where is that?
Oh, yeah.
Oh, right over here, yeah. There it is.
Cool.
Do you want to jump in? Do you want to jump in?
No. I was just coming to check and see where we were at.
Another one.
Definitely made it to
You
waypoint 9.
guys called.
I did a little
Yes.
deviation because this is now, I think. we can
Yeah, there's a
see
third one, I think.
I don't know if you've been keeping up with the commentary. We thought we summited this, but apparently there was a bit more elevation to go. So, 1892, is that the same depth that we hit last time in the
No,
first?
1895, so this is higher.
Okay, this is higher, yeah. Slightly higher than
I
the.
would say that this is the summit appears to be on top. I'll look around. You can tell that those are some-
Skate egg. There's a bunch of them up here. This could be a skate nursery. There's a fifth one.
Look at that one straight in.
Wow, that's gorgeous. It's just
Can you zoom in or turn the
Surreal,
camera?
just
a smidge so
gorgeous,
We don't have that.
view here.
There we go. Perfect.
It's a more colorful of a site than I've seen till now. So because of the what?
That's good.
The depth, yeah.
Adam, how much time do we have?
Take a little look around here.
Maria's asking for a sample.
Yeah. So this
Wow.
this one right in front is almost like the exact top of the.
Yeah, this
It's
anemone.
like, I'm the king
Yeah.
of the-
Yeah. It's my mountain.
Is that an anemone?
Yeah.
Do you want to jump on to this last little bit?
Oh, yeah.
That'd be great. Yeah.
Let's try to find somewhere with a skate egg
Skate. So
and then sit down next to it so we can look at it and then pick up the wrap right next to it. How does that sound?
But they lay the eggs. And like those eggs can take a long time to hatch.
I think there's one just back here, a little bit goes back.
It's a good chance that we've reached the summit here.
No, I was just gonna help him, but he already got the camera.
Just
They look like little leathery, tappy things. Yeah. But they've got the four corners kind of stick out like that. And so,
seen a pretty high density of skate eggs here. So we're on our
Oh, there's one.
yeah, we're searching for one, and there's one there next to that sponge, between the sponge and that coral. I believe that the goal is to get some high definition video of the skate egg, and perhaps have experts back on land determine their viability and/or species. this skate egg view followed by a rock collection should wrap up the activities of this dive.
Yeah. Yeah, there's some.
Oh, is that two right next to each other?
Yeah.
Wow.
Oh, hold on. There's a bunch covered in sediment.
Yeah. They're everywhere.
Missing all those.
They're everywhere. Again, due to the density of skate eggs here, we have reason to believe that this is a potential skate nursery.
Take some highlights.
Yeah. Do we need the lasers, Ben?
Can we get the lasers on just so we can see the size of these?
Again, once these lasers come
Yeah,
on,
the
these
one-
are two
The one
green
that's right
dots.
in front of the lasers might be a good one. How do you think about that, Maria?
Yeah.
This one? It's only 10 or 15 centimeters.
Yeah.
Not that big. Yeah, they're hard to see sometimes.
And follow-ups, okay.
What was that?
It doesn't need to go in the wire box. It can go in the rock box.
Super in on it. You can do that. Like we zoomed on a couple, but now that we're parked, we can just get one real nice close zoom on that egg.
Oh, yeah.
And yeah, like that one looks like it's in better shape than the much more faded ones. Gosh, you don't have any way to shine a light through. You can candle them. Yeah. Yeah, you can see through them. They'll candle them when they're out in the wild.
If we have time, we
Is there
can
a
go
need to
check
collect
one
one?
out.
Yeah.
Cool. We're back.
Again, for those of you that are just joining us, we are wrapping up a dive on board the research vessel Falcor 2. We have our remotely operated vehicle at the seafloor now in what we believe to be the summit of a previously unexplored seamount called Tengosed. We arrived here just a few minutes ago and were blown away by the number of different skate eggs that litter the seamount here, or the summit of the seamount here. There's good reason for us to believe that this is a potential skate nursery. What you're seeing there is Sebastian's robotic manipulating arm, and we are attempting to collect the rock sample for geological and I suppose microbiological analysis. Got just a few minutes left of this dive. If we're able to pick up this rock with not too much trouble, we should have just enough time to perhaps collect the skate egg and bring it back up on deck. Right. Yeah. Got a couple more days to explore this site as well. So our chief scientist just pointed out that we could also come back if we needed to have a more intimate look at this potential skate nursery at the summit of, again, a previously unexplored seamount called Tengosed, or I am thirsty. It's translated from Spanish. We are in the Costa Rican Pacific Ocean. How far away are we from land?
About 150 kilometers.
All right, 150 kilometers off of the port of Punta Arenas, I believe, in Costa Rica. We are joined by our Costa Rican colleagues. Half of the expedition's scientific party is Costa Rican colleagues. And it is amazing to see this part of their country. 92% of their country, I believe, is ocean seafloor. So it's extremely important for Costa Ricans to explore the seafloor, since that would be, again, exploring most of what Costa Rica is. Fantastic. Wow, slam dunk. Slam dunk.
Do
Yeah.
you have time to pick up a skate egg? Throw it in a bio box?
Do you want us to use the soft drops for that?
They can be glued a little bit. You know how the four corners are out? Like the corner corner that's lowest is most likely to be kind of attached to the rock. So if you could use the soft grip to grip that corner, that's probably the best bet on those. I mean, they're real tough though. Like I don't imagine really ripping it open too easy.
We're having a discussion here as to which robotic arm to use to most appropriately make this collection. So SCADEX are hypothesized, or I guess are known to be tough, but we do have two arms. There's two choices on what tool we choose to use to collect it. The arm that you're seeing on your screen there, it's all the, I guess has a stronger grip. The other arm that we'll bring on the screen in just a second here has a softer red lining that allows us to more easily manipulate potentially delicate samples. So we are repositioning the sub closer to that skate egg to again make the last sample collection of this remotely operated vehicle dive.
No. All the spaces used up.
Yeah.
Where will this be going?
It can go into any of the biobox chambers in the back. There are 2 or 3, biobox 2 or 3.
I guess it's that much real news.
So again, this robotic arm on the ROV working 2,000 meters below us is what we use to collect samples. And it is being operated with incredible dexterity, if I may add, by our ROV pilot team. And despite SCADEX being pretty hardy, we're trying our best not to damage the specimen. And this robotic arm on the right side of the vehicle has this softer lining that allows us to more carefully or more delicately handle the soft tissues of some of these biological samples. There it is. Okay, we've got a skate egg. Final order of business here is to get this sample in one of our biological collection boxes.
2 or 3.
2 or 3.
3's got the smaller compartment, so maybe that one's better. And thank you to whoever took out the markers from those.
Carl. How about the starter one?
Up. Okay. Looks like we got past a quiver handle hurdle there. And all right. So we have just collected a skate egg from the summit, or what we believe to be the summit of Tengosed Seamount. This will wrap up our activities at the seafloor for today.
Can you just mark that this is where we are? Yeah, but you think this is the summit? Thanks.
It's basically
Marker's just fine.
It's just this little.
Yep.
This nothing.
Yeah, just so that I remember that it's not 1.9.
Yeah. Do we begin the ascent?
We're up.
Okay.
Top bottom.
And we are taking off. Again, what you're seeing here is live video of our remotely operated vehicle, SuBastian. Just completed a 12-hour dive.
Well,
Close 10 hours.
10.
Yeah, 10-hour dive. Masterfully piloted by the Schmidt Ocean Institute ROV pilot team. That there is
narrated, Gus, on your first shift.
Yeah. So again, we're seeing a view here of what we believe to be the summit of this seamount that's crowned by this anemone there. This is the first time that humans have laid eyes on this seafloor, very large seafloor structure. We shall return soon. And we're off. We are 1,879 meters beneath the waves, and it will be about an hour and a half worth of an ascent before we break past the waves and try to recover our ROV, stowing it safely back in its hangar, again on board the research vessel Falcor 2. All right.
All right, folks. If you're still there, we've got just about an hour left before the stream has to end, when we would normally restart a stream. It won't be long enough for us to get all the way to the surface, but I think we'll leave the cameras running for about an hour and see if we find anything fun in the blue water on the way up. So thank you all for joining us on this dive, and I hope you enjoy the ascent. right. So this is Alex again. Hope you've enjoyed the ascent. We got up to about 410 meters from the surface, but the timer on our stream is running out. So thank you all so much for joining us. We are planning to be diving again, same time tomorrow. So enjoy the rest of your day or evening or morning, wherever you might be. And thank you for exploring with us.